स्क्रीन शेयर हो रही है सर, देन वी स्टार्ट सर.
So you also send the link on mail?
Yes sir. Pani sir, screen is shared.
प्रॉपर हो जाने देना महन जी है.
जी सर.
Sir, PPT shared on YouTube, sir.
Let me check sir. Yes sir, PPT is shared sir. So with this sir,
Sir.
Mujibuddin sir, screen is off sir.
Yeah, sir. I am pinning myself, so for that purpose, I have stopped this. So I am sharing my screen, sir.
So, welcome you all. Good morning, one and all present over here. Today is the day dedicated to the celebration of Shri Guru Gobind Singh Ji's Jayanti. On this auspicious occasion, we honor the life and teachings of 10th Sikh Guru, Shri Guru Gobind Singh Ji, who inspired courage, equality, and justice. His unwavering commitment to righteousness and the empowerment of individuals continues to guide us today. As we celebrate his legacy, let us remember his values of selflessness, unity, and devotion to the greater good. Dehe Shiva Baru Mohi Ihe, Shubh Karmante कबू न डरो न डरो अरसो जब जाई लड़ो निश्चय करी अपनु जीत करो. That means, O Shiva, bless me that I may never hold back from performing auspicious deeds. When I go to the war, I should not be afraid of the enemy and ensure my victory in the war. Myself, Dr. Kamya Goyal, Professor, Department of Pharmaceutical Chemistry and Analysis, ISFCP Moga, warmly welcomes you all. We are gathered here on Day 4 for 6 days outcome-based faculty development program on empowering faculty through enhancing teaching skills in pharmaceutical education, organized by ISF College of Pharmacy in collaboration with IKGPTU Jalandhar, APTI, and IQAC-ISFCP. I heartily welcome our eminent director, Dr. G.D. Gupta sir, Dr. R.K. Narang sir, Principal ISF-CPR, Dr. Siddharth Mehan, IQAC Coordinator, ISFCP Moga, worthy speaker, and dear all participants. Today, on day 4, we are having online lecture sessions. Our first online session chaired by Dr. C.S. Mujibuddin. Dr. C.S. Mujibuddin is a highly accomplished professional in the clinical research industry, holding an M.Pharm and MSc in clinical research from Manipal University. With over 15 years of experience, he has worked with leading organizations such as QPS, Perexel, Oracle, IQVIA, gaining extensive expertise and making significant contributions to the field. Driven by a passion for teaching, he founded Clinosol Research Private Limited, where he has successfully trained and mentored over 1,000 students since 2019, preparing them for successful careers in clinical research. Under his leadership, Clinosol has gained recognition, winning 9 prestigious awards for his excellence in skill development. In addition to his educational efforts, He actively participates in industry conferences as an academia partner, contributing to events like ISCR, SCDM, etc. His commitment to education, innovation, and industry advancement has solidified his reputation as a leading figure in the clinical research community. His topic for today's lecture is Revolutionizing Education, Old Teaching Methods versus AI-powered Innovations. I would like to invite our today's first session speaker, Dr. C.S. Mujibuddin. Sir, over to you.
Yeah, thank you, sir. Thank you so much for inviting me, and thank you, Director, sir. Thank you, team of ISF College of Pharmacy. So I'm very fortunate to be here, and I'm happy to share how the education system has evolved from old teaching methods to new teaching methods. So I'll just wait for a second. If I get a heads up that it is live on YouTube, then I'll start my session. And if my screen is visible, I'm audible, I'm visible, then let me know. If any of this not is done, please let me know so that I can wait for team to confirm. So I'll wait.
Parneer sir, can you confirm?
Yeah. So if my screen is visible, I am audible, everything is clear, then I'm ready to go ahead.
Yes, sir.
Okay. So can I take it as yes now? Okay. Can I take it as yes? Can anyone confirm me?
Sir, wait a second, sir. Parneer
Yeah.
sir, please confirm, is it live on YouTube or not?
Yes, sir. Yes, sir.
Yeah. Is my screen visible and I'm audible and I'm visible?
Yes, sir. You and your
Yes, sir.
screen is visible.
You can continue, sir.
Yeah. So now today's topic for the session is about how education system from old teaching methods to AI-powered innovation has taken care. And definitely AI has part of our industry, and definitely pharma industry also is growing so much with respect to technology. So here is my agenda. I'll start working on how the old teaching methods were there. Like it means that a classroom where you have a whiteboard or a chalk piece kind of a teaching methods, and the newer teaching methods powered by AI. How AI has taken care of some of the trainings and how we have tailored some of the education systems. And then I will be comparing the old teaching methods with the new teaching methods. And I'll be giving some of the case studies, like how AI has implemented, and those case studies with examples I'll be giving. And what is this impact on the teachers and students with respect to AI implementation in teaching? So that's something which I'll be speaking today. And finally, I'll be putting something which is called as conclusion with respect to this. So can I request the ISF College to make me as pin so that my video is visible? Can someone from technical team can pin my video so that my video is visible in YouTube also? Okay. So a journey from traditional to technology. So if you see left-hand side, if you see the image of left-hand side, how it is being shown is it's a classroom where students were teaching with books and whiteboards. And right-hand side, if you see a person with a blazer who is sitting on a laptop or a tablet, and people are working on a laptop or a tablet with analytics kind of things. So this is the journey from traditional to technology, and how the educational system has changed, and properly in pharmacy sector, how the traditional to technology has evolved. We are going to look at it today. So now first one, imagine a classroom wherein the first picture shows that how the classroom was there. This classroom initially was properly designed for an activity where they have one classroom and teacher is standing, and it's a universal syllabus which is being taught. Students will come with an empty brain. They'll not learn anything before they come to class, and they'll come and sit, and teacher will start explaining about the processes, the maps and all those stuff, and students used to make a note of the books. But now going forward, what might be the futuristic learning is something this picture, how the AI is being dealt to the students who are sitting with zero paper, zero pen, and with respect to technology. So the technology driven classrooms or the futuristic classrooms. So now a century ago, it was rows of desks and teachers lecturing in front of them, say, putting in a whiteboards and all those things. And there were notes and pens and all those things. And there were not these three words: decentralization, decarbonization, digitalization. Now, all these three words were not there in the first picture. It means that every student have to come to a classroom. That's a centralized one. And every student used to write in a paper and pen, which is wasting your paper by cutting the trees. And the last one is you don't use anything which is soft copies. Everything is hard copies and paper, and you don't archive them. You throw the papers at the end of the year or something like that. So decarbonization, digitalization, and decentralization was not there in the picture 1. While in picture 2, if you see, all these three words are there. So it was a decentralization, decarbonization, and also digitalization. Now, in picture of a smart classroom, there might be interactive screens. Like, say, for example, the screen is visible for the teacher. It might be visible for the students, and they might give the answers. Now, assume that we don't know what student is understanding or not. And we started a class with A, and we ended up at the Z, and assuming that students have understood. And teacher is still assuming that I'm the best teacher and students have understood. Rather than this method of a historic method, why can't we take a step back and say that every 10 minutes, I'll put some poll questions. And let's see how many of you guys have understood my system or my teaching methodology. Based on it, I'm changing my pace, I'm changing my assignment, I'm changing my assessment, I'm changing my case-based scenarios. Every 10 minutes, put a poll questions where every student is answering the poll questions and then answering it in a proper way. So that's the second one, which is your updated modern classrooms, which is interactive screens, which speaks with you, AI-driven assistants like chatbots will be there, who can give you more learning and collaborating virtually across the globe. It's not about learning at one particular zone, but across the globe. Say, for example, I'm at Hyderabad now, but I'm giving session to across the India or also world. All the teachers who are putting here efforts were across all kind of areas who have joined today. So now this is something which is the futuristic learning and futuristic area. Okay, so now what is AI? So how AI is shaping teach, learn, and connect. So there are three words. First one, teach. You teach to the student as well as you teach to AI. And you teach to the student and you teach to the AI. And learning is for the student and also learning is for the AI. And also the AI is connecting with the student and learning more and increasing this. One size does not fit all. So say, for example, earlier days we used to say, okay, this is a universal syllabus. Everyone have to undergo the syllabus to get your marks and percentages. But one shirt does not suit for everyone. One medicine does not suit for everyone. And one syllabus does not suit for everyone. One pace of teaching does not suit for everyone. And one learning methods does not suit for everyone. So now this is something which we have to understand, that dynamically how I can start teaching to the student based off understanding. Say, for example, in a classroom, there are 100 students. We cannot assume that all 100 students are so much fast that whatever I am speaking, they are understanding it. There might be 0 to 10%, there might be 10 to 20%, 20 to 30%, 30 to 40%. Based on them, my syllabus, my learning, my assignment, my assessment, my project work should modify it. So now one system does not fit for all. How can we shape them based on their outcome? What outcomes, what answers they are giving, based on it, how we are teaching them is something which is futuristic learning of AI. So now a simple answer is one system does not fit for all. So now if you see here, today's focus, how we are going to look at AI powered innovation and how it is impacting on students, teachers, and future of learning. See, we need to understand that, yeah, definitely systems have become a part of our life. Laptops and technology has become part of our life. And now most of the times when students go for interview, they are not asking about percentage and marks. They're asking more about how are you dealing with situational based aspects. Because the one which AI cannot drive it is innovation, teamwork, leadership, sales, marketing, and technology oriented and teaching skills. Now this cannot be taken by AI. How are we working on this activity? Because AI can teach A plus B is equal to C. But the other things a human being has to work. How are we going to look into it is something which we are going to look into today's topic. Okay, so now old teaching methods, as usual, structured, consistency, offering solid foundation of learning. So it was very structured, syllabus was there, agenda was there, teachers were there, and everything was structured, and outcome was everyone have to pass in a particular exam. But how modern AI has provided. So it's more on creativity and also more on personalization of individual learning systems and allowing students to learn on their own pace, exploring new ways of thinking. Earlier days, I used to just work for marks and percentage. But now, future might be more of creativity, personalization, that a person wants to go into this sector, or a person wants to learn at his own pace. He don't want to come to class at 10 o'clock. He might want to learn at 6 o'clock to 10 o'clock, and then 10 o'clock to 1 o'clock, he wants to work on project work. He wants to work on internships, something like that. So now exploring the new ways of thinking, how we can do it, because human being is more active at early mornings or nights. He might not be active during the days. So that is old teaching methods, how we have done this for years together. So now coming back to my old teaching methods, teacher-led sessions, direct instructions were there. So there were teacher who used to stand on a board, and teacher was leading the sessions, direct instructions to the student. A plus B is equal to C. This is the pharmacological action. This is the structure, and this is how it is outcome. And same approach for everyone. And tools were blackboards, textbooks, and papers and pens. Now pros, what are the advantage? Clear expectation. Foundation was very strong. Teacher was there to support you every time. And discipline was there because in classroom you maintain the discipline. But there were cons also, that creativity was missing. So there was no creativity because you have already put so much of pressure saying that, okay, your agenda is this, your marks is this, your percentage is this, these are the things, these are all those things. And why one size does not fit for all. So it does not cater individual needs, that a person wants to become a poet or XYZ. It does not create that fits for all. And passive learning is not there. So now say, for example, just student listens, but he don't open his mouth. He does not talk it. So he does not ask questions. He does not engage with the teacher. So passive learning was zero. And slow changes were there. Hard to adapt quickly new methods because they don't want to come out of the classroom and sit in the technology room saying that, okay, let's teamwork on this. I will put a Zoom link and all three people together will share our screens and discuss about it. So these were the cons and pros of old teaching methodologies and old systems. And new technologies. So this is one of the best thing which I always learned it. New technology methods is, first one is powered by AI smart classrooms. So this smart classrooms, teacher will come and flip side learning will be there. It means that teacher will put an assignment to the students one day before, saying that, okay, complete this assignment, come back to the class. And student has that ease or the fire inside him that he has to learn it. And he will learn, he will come to the classroom. And during the classroom, there will not be a class, but there will be a discussion forum where student will that, boss, you gave assignment, we understood the assignment, we worked on it, we cracked our brains, but we have not understood ABCD point. Can you tell us? The teachers start discussing about this points. So that's a flip side learning. So one day before the syllabus topic is being given to the student learns, come back to the class. So interactive displays will be there. It means that one student comes, he gets the question there. Put it a question saying that why everyone have answers. So group one, why you answered this question as this? Can you justify what is your answer for this? From there, learning starts for them. And real-time participation because they are engaging themselves with a flip side learning, and also they're collaborating as a team and answering that. It's not one-to-one question, it's situational based questions. Now, say for example, if I ask one-to-one question, what's the capital of India? Delhi. So that's the answer. Days have gone where hours together teacher used to teach and student used to listen, but now it's only 20 minutes of time. Now split the sessions in a very shorter time. 5 minutes of session, 2 minutes of poll, 2 minutes of break. 5 minutes of session, 2 minutes of break, 2 minutes of poll. Like that, you have better retention, better focused learning and lessons. So micro learning is chip based, short learnings, and then you start learning on it. Then very interesting name, project based learning, which is most interesting one, that you give a project to them. Say for example, we are speaking that India is becoming capital of diabetes. Now, what is the proper reason of diabetes? Can we take the surveillance studies in a particular zone, that how much fats, how much proteins, how much things are being eaten by youngsters, how much screen time is there, how much walk is there, how much sleep is there? Can we do it? Is something is project based learning. So we can do a surveillance studies and we can solve the activities with respect to project based learning. Now, one more time, what are the new teaching methods leverages? So now I have spoken about flip side learning. I've spoken about IoT, smart classrooms. I've spoken about project based learning. I have spoken about gamification. I've spoken about virtual learnings. I've spoken about adaptive learnings. I've spoken about different kind of LMS and all the systems which can be used for the new teaching methods, what can be leveraged. Now, these are student-centered, focused on personalized learning. It means that one teaching system does not fit for all. Every student is different. Every student is engaging with the teacher. Adaptive, AI adjusts your lesson based on your progress. How you are answering based on your understanding, AI is adjusting course. And it is giving that much course only. So now, the next one uses data. All existing data, that how many students have joined, what are the percentage, what are the marks, what are the project works, all that data, they progress and improve the learning. More difficult. You record the sessions based on recording how many people are giving answers. And integration of smart technologies like chatbots. We need a chatbots instead of teacher. Say for example, student got doubt at 12 o'clock. How can we develop a chatbot where student can directly go and talk to chatbot saying that, boss, I have a question. Which class you are? Final year B Pharm. What is the subject you are looking at? Pharmacology. Then what chapter? This chapter. What question? This question. Then that chatbot will answer all those things. Now that's something which we can develop by our systems, chatbots. Learning apps, everyone should have their learning apps. And real-time feedback, instant feedback, you will get the help to student to improve very quickly all those activities. So now this is the advantage of the AI systems used in your activity. So now the most important, the impact on teachers and students, how it is going to impact. See, definitely it is going to change the way we are looking at teacher. Okay, so teachers might have work reduced, but with more automations, like grading and scheduling the classes for individual students and classes for individual activities, more of recorded sessions, more of activities which can be done. And insights from AI-driven data analytics, how AI-driven data analytics to adjust the teaching strategies, how we can adjust the teaching strategies, and upskilling is needed for the teachers to understand LMS system or flip side system, gamification system, virtual assistant system, or adaptive learnings. More knowledge is required with respect to data analytics, R and Python programming for the teachers to understand how they can improve their own chatbots and chat systems. And student, the personalized learning pathways tailored for individual needs, how we can put individual needs, and flexible learning, which is 24/7 student can learn, and higher engagement. Student can engage with teacher like anything. virtually or directly. So how we can put gamified methods. So this is something which is impact of teachers and students. Now future education, how it will be? It will be blended of learning, which can be AI powered tools, seamless learning for the student 24/7, any point in time. He can connect with chatbots or teachers and other things. And focus on creativity, critical thinking, and human connections. More important for futuristic learning. AI trends will be there, continuous learning, upskilling throughout the life. It can be hybrid education. I'm not saying completely AI system. It can be sometimes one-to-one and hybrid systems, and AI-driven insights with data analytics and data visualization and data science for the future of learning. So this is something which is more important for the future of learning. So now comparisons, the old education to current education, the personalized engagement, it's a minimum at old systems, but new methods, it's more high. And now engagement, earlier it was very limited, but now it is more high interactive, and we get the student what he wants it. The tools and resources, blackboards and textbooks earlier, but now AR, VR, and AI, LMS, gamification, something like that. Flexibility, it was fixed situations and location, but now new methods, anytime, anywhere learning. Scalability, teacher is dependent only to train 100 students, but now it is automated and scalable to lakhs and lakhs of students. Assessment, standardized test, but real-time analytics can be used with respect to AI-based teaching systems. Now, the overall conclusions, AI transforming the education by making learning systems more personalized and engagements, and also combining the traditional methods with AI tools creates a balanced approach. The balanced approach meets the need of the students and how we can use the AI tools human to bring more of creativity and imagination and emotional connections into your educational system. If you see a picture here, which clearly states a person who is connecting with all kind of system. The future lies in combining of AI efficiencies with human imaginations and reshaping the meaningful innovation education system. So this is something which is more needed for the current teachers, how the teaching has been grown now current industry. So I'll park this now for a minute, and then I'll take any questions so that we don't miss the engagement of the session. So I hope my voice is audible, sir. I hope my voice is audible, and my
Yes, sir.
yeah. So I will be stopping. I will be holding here for a minute so that I can take questions regarding the newer education system and AI-driven education system for the benefit of students and teachers, how it can be implemented and how we can go for it so that I can have some engagement. And my second part of session is how AI is revolutionizing the pharma and healthcare industry, how AI is transforming the pharmaceutical industry, research and development and clinical trials industry. So how we are going into a right directions into it by using the powerful applications of data science or AI or data mining or blockchain technology, all those terms we are looking into it later on session. So I'm open for questions, sir. If anyone is posting some questions, I'll be happy to take a couple of questions and then we'll continue our sessions with some questions.
Dear all participants who are joined online, as well as the faculty members of ISF College of Pharmacy, if any query related to this topic, so you can ask to Dr. Mujib.
Yes. And if you ask more questions, I'll be happy with more questions so that we understand that the sessions what we are continuing is going in the right directions. Sir, you can take some questions if you get any questions in the chat box or from the Google Meet or something like that, sir.
Definitely, sir.
Good morning, sir.
Good morning.
Sir, in physical classes, there are lots of ways to teach a student in a practical way. They can learn the practical skills. Is any alternative available in AI? With the help of AI, we can teach the practical skills to the students.
Yeah. So now the question here comes is the practical skills while you are teaching in a classroom, can you teach it in the online? So that's what I am saying. Simulations are already being developed. So now say, for example, I'll take a very vague example, that HPLC. You wanted to showcase the HPLC, how it works in the lab. Then all 10 students have to travel, come, sit, and then you have to show the HPLC. Rather than doing this, can you record a video of 360 degrees of how HPLC works in the system and put it on AR and VR for the students, where students learn from home and they directly come to class, start doing their lab without wasting this 1 or 1.5 hour of time for traveling and other doing things, and remaining things they can do it. See, the point here is, we are not saying that all the activities will be lost, but 40% of activities can be reduced for the student, and remaining 60% we can do it with physical appearance also. And we cannot say that AI is going to change all the education system in one day or something like that, but definitely it is going to get changes. Now a student is understanding VR and AR. Tomorrow he wants to get a project from US. He has an idea that, okay, I can propose my project to US also by using AR and VR, where my sir used to teach me this systems. We can do that. So definitely the practical approach, one-to-one teacher is needed, but we can revolutionize the activity of AI taking into the pharma and healthcare. So I hope I have answered that question.
Sir, another question by Dr. Balak. So I request Dr. Balak, please ask your question.
Good morning, sir.
Morning.
Sir, can you suggest some freely available AI-driven material helpful for teaching?
Yeah, so the slides, I'll definitely give it back to the systems. And there are some systems which I'll put it later on into the group, or I'll send it to sir. There are some systems which can be learned freely available. And basically, AI is not anything which is different than digitalization of systems. I'll tell you something here. Now, say for example, you develop your own system. You take a class of pharmacology of cardiovascular drugs. Okay, so now this class is of 2 hours. How can you drive it through AI is something which we can answer. Pharmacology of cardiovascular drugs. Okay, so now first of all, the background information and heart action, and then what are the different drugs, what are the different mechanism of actions, what are the efficacies, what are the adverse events. You mark it in a way that you understand it clearly in a structured way. Then student will be given with the sessions individual parts. Now, say for example, you take one session and give the session to the students, either through videos or through audios or something like that. And then you develop questions, poll questions, and you give the questions to the students. And questions are being answered by the students, and they will get back to you based on how they are learning. Based on it, you are modifying it. So 2 hours of class, you are making it as 4 hours, but with 100% of engagement, they will not forget in their lifetime. So that's something which is needed for us.
Okay, thank you, sir.
Yeah, thank you.
So,
any other questions anyone have?
Next question, Dr. Manish.
Good morning, sir.
Good morning.
My question is, what are the limitations of AI teaching?
Limitation is your physical emotions and mental emotions might not be there in teaching. See, if I come to Punjab, I'll be very happy to travel, and I wanted to come and give session in front of everyone. But the aspect is, you save the time, efforts, and everything, then that's the biggest problem. So the physical activity and the emotional touch between the teacher and student. The AI is revolutionizing definitely, but the earlier times, the relationship between the teacher, the guru, shishya, kind of a relationship between teacher and student was missing. And the ethics, values, and respect to the teachers might be a little bit here and there, because we are more of technical things. We want to teach them technically, creatively, project management, leadership, and other things, rather than putting ethics, values, privacy, rights. All those aspects might be missing with AI-driven teaching. The futuristic learning might not be the historic way of learning, that how a student should behave when they go into real-time industry. So I think that's I feel like that's something which is more. See, the students are not ready to accept what is wrong and what is right.
Right.
So they think that wrong. Say, for example, movies have impacted us so much that a hero might kill 100 people, but still he is a hero. So that's the impact movies are giving on the youngsters. The teacher is the only one person who says that wear good dress, behave nicely, go and touch your feet of teacher, go and respect to your elders and parents. Teacher is the only one who is teaching this. But with the impact of AI, I feel like that somewhere we are missing that point. And we should implement that points, and we should have some trainings of ethics and values and rights for the students, that how they can learn. I hope I have answered your question.
Yeah. Thank you.
So
Thank you, sir.
Next query by Dr. Khadagraj.
Good morning, sir.
Morning.
Could AI replace the traditional teachers in the future? What do you think about this, sir?
Yeah, see, the basic agenda, I'll tell you. The story here is AI is going to take our jobs. Answer is yes and yes and yes. But here, the answer one more time, if I repeat it, AI is going to create more jobs. Answer is yes and yes and yes. So now AI is taking the traditional jobs, definitely yes. AI is going to create newer jobs, definitely yes. Now you have to understand, you have to touch base AI, implement latest and innovative and other learnings, or else you are not going to survive in this world. So AI, you have to understand how you can digitalize the technology, gamify it, learning management system, adaptive learnings, and also flip side learnings, macro learnings, all these learnings, how you can split it, make it more interesting. Then your jobs are safe, your teachers are safe. But definitely teachers are going to have hardship or hard times, because now OpenAI is going to bring one more tool, which is automated video tool. You can type something like, say, for example, pharmacology of aspirin. If you type it, it gives a wonderful video of 1 hour. But the problem is it can give the video, but the level of understanding for the student is not sure. Student might listen for 1 time, 10 times. But the way you teach cannot be changed. Human being, how he teaches, how he engages, how he makes the things very easy for the student is more important for you. So that's the beauty, that teachers cannot be replaced, but teaching methodologies can be replaced. Now you can't say that I don't want feedback. I don't want to listen to students. I don't want students' happiness. You need to have quality of index of a student happiness about the learning and outcome, then only you can survive in the industry. A teacher's life might be in future, it will be outcome based of the student happiness. So it's all about feedback system, data analytics, data visualization, data science, and data mining. Then only we can put our steps into the future. I hope I have answered the question.
Thank you, sir.
There is another query in YouTube online chat box.
Yes, sir.
The person is Mr. Jabir Singh, and Jabir Singh is asking, AI teaching methods do not have emotional connections, as we have seen in COVID online classes, and there is no discipline in students. What is your opinion about this?
Yeah, see, definitely I have already said this very clearly, that AI might give some challenges. But the problem here is there are solutions also we have to get. And I'm really sorry to say, as a teachers, are we doing our homeworks to do it? Answer is no. As a teacher, we want students to listen disciplinedly. But students are not the way earlier students were there. Now we need to modify our gears, and we need to make sure that 80% of students are good. Remaining 20% are worse. Forget about 20 students. Only focus on 80 students, giving assignments to them before they come to class. You go and tell them, okay, you prepare this presentation. Tomorrow, 5 minutes, you give presentation. The student number 1 will come and give presentation. Remaining all 79 students will get activated. The emotional touch, you have to create it, or else they will not do it. So now, to make teachers to ask questions, I stopped my session and I engaged with the teachers. Why can't a teacher also do the same situations? That how can you engage with the 80% of people, remaining 20%? Because always Pareto analysis is that 80% are good and 20% is bad. Don't focus on 20%, because emotional touch is 20% is not there. 80% are good, save them. Discipline is still there. They can unmute, they can talk if they like you. if student likes the teacher, it's a no doubt that they can do their presentation, they can do these activities. See, recently I have conducted a couple of workshops in a couple of universities, and every student have submitted their presentations, their project work, their case reports. And the teachers and the deans of those universities were really shocked that our students are able to do this all activities we don't know. So how can you engage them? How can you make them interesting that you can do it, you will do it and do it? Rather than just saying that you have to get marks and you have to get percentage, you have to read this, by heart this, do this, then see, there is no system called as by hearting now going forward. The ChatGPT is already so much by hearted system that you just type it and you get answer. What is not there is innovation, creativity, leadership, project management, and data science. So this is something which we have to get it into this system. I hope I have answered the question, sir.
Yes, sir. Sir, there is another question by faculty Sakshi
Yeah.
Gupta. Ma'am,
Yes,
please.
Good morning, sir.
Good morning.
Sir, my question is, as per your opinion, what is the best AI tool which we can use in our classroom in the initial stage?
So I hope initial stage I would like to tell that flip side learning is one, and poll questions is the one more. Poll questions and flip side learning, and mark your students every week one day zero paper policy. Implement it.
Okay.
Zero
Okay,
paper
sir.
policy that students should not bring one book also, one pen also. They have to bring their tablet or the laptops and show it on the system. So their project works, their activities, it should be so innovative that you don't tell them anything. You don't give a project work, you don't give a case report. You tell them, you take your dummy data, you take your case report, you talk to your friends and teamwork, and then come up with the solutions. So that's the best thing which we can start with. So my workshops planned in such a way that how zero paper policy can be implemented. So the future is all about technology and AI driven teaching systems.
Thank you, sir.
Thank you. Sir, can we start now the other session, or should I take one more question? Last question, any last question, sir? Sir, any
Dr.
last
Khadag-
question, sir?
Dr. Khadagraj.
Any last question I can take, or else because I have only half an hour of time, then
Yes,
Ravishankar
sir.
will
Please,
join
please
us.
continue, sir. Please continue your talk.
Yeah. So now, more interesting technical session. Now, till now we spoke about how teaching can be done, but how AI has revolutionized the pharma and healthcare industry. See, now the basic agenda I'll tell you that how AI has revolutionized the things from individual patient to a networking circle. I'll just tell a small story that earlier days, patient used to get a disease, and then, for example, he used to get a scar or ulcer on the hand, and he used to run to the doctor and wait for the doctor, and doctor used to give some lab reports. He goes for a lab, and then he gives a blood sample, he gets the lab report, doctor sees that, gives a prescription, patient takes the prescription, go to a medical shop, and then he gets the medicines, and then he takes the medications and gives the report to the doctor. So this is the earlier story. But now story got entirely changed, that how AI is getting changed with respect to patient. Now patient gets a one ulcer on the skin, and then he opens the system, and he opens the healthcare app and saying that, okay, I got an ulcer now. And then chatbot will say that, okay, nearest doctor, who is the nearest doctor? Chatbot will book the booking for the patient. And immediately doctor will get a message that a patient is waiting for you. Patient will switch on the camera, doctor will switch on the camera, and patient will start saying, good morning, doctor, I have a problem. I have an ulcer on the skin. So this is my ulcer. My previous history is I have ABCD disease, and I have my social history. I smoke, I drink, I sleep, and I booze and all those things. I walk for this many hours, I sleep for this many hours, my stress level is this, I'm on this medication, and I got an ulcer. Doctor has taken all the data, he has put all the data into system, and system gives the data science into this and say that, okay, go for this lab reports. Doctor is getting this message that, okay, based on all this existing data, go for this analysis. And then doctor has reported, doctor has sent one lab test to the nearest lab of the patient and also one report to the patient. Nearest lab person runs to a patient and then takes a blood sample, go to a lab, analyze the lab report, give a one report to the doctor, one report to the patient. One more time, doctor puts an app saying that patient, please come on online. Patient comes online, doctor comes online, and then doctor says that based on your data, which is previous diseases, previous medication, your physical activity, your mental stability, and your allergic conditions, and your lab reports, and your signs and symptoms, the data science model has explained that you can be given the treatments, option A, B, C. Which one you wanted to take, you explain to the patient. Patient will say, okay, I will go with the option 1 treatment based on your explanation, what you have explained to the disease. And then patient will say that, okay, this treatment. Then doctor immediately put a prescription, online prescription, send it to the nearest medical shop. The nearest medical shop opens a drone, put the medication into drone, map the circle for the patient home, drone goes into patient home, puts into patient home, and then patient scans the QR code, and then he opens the medication, takes the medication, eats the medication, reports it to the patient online. So this all story, zero travel for the patient, zero travel for the doctor, and zero expenses, money, efforts, but highest engagement. Patient never miss the treatment. Now here, what else we have included? First thing, we have included telemedicine. Second thing, telepharmacy. The third one, data science models. And the fourth one, outcome-based patient centricity. Patient is given decisions. And the fifth one, drone-based dispensing. The sixth one, blockchain technology to give the payments, the QR code payments. And the seventh one, the outcome-based based on the patient feedback. All the technology is rotating and revolving towards the patient. How can we teach these systems to the students is something which we wanted to understand. Why can't students develop their own data science models to give the treatments or to give the drug interactions and to make sure that how it is going to happen is something which is AI/ML in pharma and drug discovery. So now in drug discovery, so there are three things. Most importantly, first one is in preclinical, in silico screening is already there. There is already machine learning algorithms which have developed so much that virtual screening can be developed. How much it is getting connected to a receptor, affinity of a drug to a receptor. There are 100 drugs you have, but we don't know which drug can be given. Affinity to the receptor is something which is needed. And in silico model, virtual screening we can develop, algorithms we can develop. Very recently have done one project work with one of the university, and they have developed a model of a drug discovery. They have put some 10 drugs, and they don't know which drug is more having affinity. And virtually you can do a screening which is in silico screening, and potential drug candidates can be identified very, very rapidly. The next one is predictive modeling. So predictive modeling is something which can predict the drug efficacy, toxicity, PK, PD, and also make sure the drug discovery makes very quickly. Future is not about drugs. Future is about biologics, vaccines, devices, cosmetics, and nutraceuticals. Future is not about the medications. Future is all about the biologics, devices, and I think very recently, I think Cipla or some company, they have got, yesterday, if I'm not wrong, yesterday they have got one new route of administration for insulin. So that is a blockbuster. So the future is about how the medication can be given in a different route of administration than just a tablet or a capsule. So that's a predictive modeling. The next one, clinical trial optimization. How can you take a design? You have a drug, you have a disease, but how can you prepare your own protocol? By putting into a library system. We have hundreds and hundreds of protocols, put it into a library. You go to a library, you say that my drug is this, my disease is that. Can you give a design? So the library will analyze it, data science it, and then they give the protocol design. It might not be 100%, but 60%. Yes, definitely. Patient can be selected by using webinars. Patient can be done by using the EHRs and EMRs, how they can do EHR, EMR things by patient selections. And we can do data analysis because so much of data is available for the patients. And we are sitting on data lakes. Now, say, for example, I have Telangana and Punjab. So I want to know which state has more cold cases or fever cases during month of December and January. Based on it, I want to identify which paracetamol should be given to Telangana or Punjab. Data analysis. We can do a lot of analysis with respect to clinical trial optimization also. So these are the three things which can be developed in the future. And yeah, definitely the future is on personalized medicine. I have already said that one shot does not suit, one teaching method does not suit, one medication does not suit. How can you make sure the patient, see for example, paracetamol 650 mg does not suit for everyone. Not necessary that paracetamol suits for everyone for this. How can you make sure the AI algorithm analyzes the genomic data and give that, okay, patient has this specific gene and this drug can act on this gene, and also you need this much dose and this much things for making your adverse event and efficacy. That is what is called as genomic analysis. And the next one is stratifications. You can stratify. You can't say that one drug paracetamol can be used for all the patients. One male, one female, age group, gender group, race group, particular state group, all the groups, how you can stratify. Because based on biological, based on the stratification, based on the gender, based on the race, the drug efficacy, safety can be changed. How can you stratify? And the last one is treatment response prediction. The future is not paracetamol is reducing fever or not, but on paper, how the treatment response prediction can be understood is something is future of your AI. So this is something which is very, very interesting stories the AI can be doing. This is very interesting story. The future is about medical imaging and diagnostic. There might be the future where you don't need an X-ray and scan or ECG image to be taken to a doctor, because we have developed already an algorithm and we have given enough images, X-ray, ECG and scan reports and everything, and we have taught to the AI system. Computer aided detection for the diagnosis of the images. The conversion of images into data and understanding the data science of the diagnostic is something which is medical imaging diagnostics. The future is you get a scan, you put it a scan into the system, and then the system will say that the proposal is disease A, B, C, D, for example. Doctor might not spend much time to diagnose a particular disease. That's what you call it as computer aided imaging diseases technology, where they can understand about images, how it is going to happen. So it's all about data and data science. Yeah, definitely there is something which is clinical decision support. Now, patient comes to a doctor. Doctor cannot just give a sign and symptom treatments. First, doctor has to understand multiple sources from his data. Say, for example, he used any medical shop bills, insurance, any alcohol, any smoking history, any walking, jogging, all the variables, sensors, phone pay records, and insurance record, everything, EHR, EMR, every record he will have. Who? Doctor. And doctor will analyze all the data, and then he start giving a recommendation of a treatment. Once he started giving a recommendation of a treatment based on the machine learning, the evidences which are being given the patient will be one more time continuously monitored, and then the next level of treatment will be given. Now, say for example, earlier days, we used to say that if you want to see anything in the stomach, you need to send a pipe into the stomach and then do it. But now, recently, AIG hospital has started a clinical trial on a capsule where you have a camera in capsule and you go and put it in the stomach, and then capsule comes and camera comes and takes the pictures of the stomach. You don't need to put any invasive process to it. So continuous monitoring by using smart pills, or by using patient data, or by using alarms or e-pros or wearables or sensors, all these things we can continuously monitor, and we can continuously do these systems for the patient respective treatment. So this is patient respective treatment. We can give this type of teaching to the students. Yeah, so this is one more time very important, that we don't need drugs, but already existing drugs, we can repurpose it and we can optimize it, because you have already drug which is available for years together, and you don't need something big problems to be happening here. So it means that the safety is already established, just you have to establish the efficacy. That's the biggest advantage of drug repurposing. Focus on drug repurposing and optimization of the computational chemistry and neural chemistry and genomic analysis. All three together, data science, how you can utilize the machine learning and AI for the drug repurposing and optimization is something very important for you. Now, next slide. Yeah, this is very important, patient engagements and monitoring. So earlier patient used to come and speak, but now patient is not coming. How you can monitor remotely patient monitoring? AI analyzed algorithms for wearables and sensors. Now people are using watches, people are using rings, people are using different kind of sensors, connected devices. And also they are using some healthcare apps also on phone, where if patient says that I got this problem, then AI chatbot itself is giving answers to the system. If there is an emergency, then AI chatbot is directly reporting to the healthcare systems or AI or the doctors. And virtual health assistants, which is ChatGPT or chatbots. AI powered chatbots. Students can be encouraged to prepare their own health assistant apps. Particular disease, you can develop apps. Particular conditions or doctors, you can develop apps. AI powered chatbots, where patient can directly go and ask chatbots, voice assistants, to provide information of the drug and other advertisements to the patients. You can give virtual assistants, and also you can give reminders and support to the patient by using chatbots or virtual health assistants. And medication adherence. The biggest problem with current patients is they don't adhere to doctor. They don't listen to the doctor. You can put how many medications have been given and how many medications are being adhered, and personalized interventions can make sure the interventions are improving the compliance of the patient drugs. Engagement is more important and monitoring is more important, but with the use of AI and digitalization, how are you going to increase it is something which we are going to look into it very carefully. So now robotics, this one is more important for us. Robotic surgeries and rehabilitations. The rehabilitations, I think, more interesting. There are more of AI robot systems which are coming, and the rehabilitations are more with the physical therapies, and also the physiotherapies can be given with the robot systems. AI-enabled robotic systems can give more assistance for physical rehabilitations, where human being might not be able to give physical rehabilitations, and personalized therapies and also tracking the progresses it cannot give, because human being might forget the data, but system will not forget. The robots can give you more rehabilitations, and surgical aspects also. Robots can give more activities on the system. Yeah, so this is more interesting, supply chain. So earlier days we don't used to focus more of supply chain, but now future is on all supply chains. How we can do supply chains. Now say for example, last year, 10 years back, I have had a data. So every year, how many people are using paracetamol in particular place, or how many people are using antibiotic in particular place. If I have that data and analyze that data, then I predict another 10 years how much data I wanted. Then you can improve your supply chain in a particular time zone. You can create demand. You can look at how inventory can be managed and how you can make sure your logistic is being done. Your travel, your storing, your inventory, and your activity can be done. You can do with the supply chain optimization with the use of AI, particularly blockchain technology. And one more thing, we can save the paper. Say for example, in a box of a paper, in a box of a tablets, you get a big label of paper. Why are we getting that paper? Can we have a QR code on the tablet itself, and you take a phone and then scan it, and then you get all the labels? How can you create the demand for particular disease? How can you say that what is a futuristic disease we are going to get in future? How can you improve the medications? How can you improve number of patients and improve the number of patient outcomes is something which is supply chain optimization, wherein every product, every drug can have QR code, and patient can understand whether this drug is real from the particular company or not. Currently, we don't know whether it is Dr. Reddy's or anything. How can you know that it's been manufactured by Dr. Reddy's only? It can put a drug, Dr. Reddy's name, and they can put a batch number. We don't know. So for that purpose, you can use a QR code, and that QR code will give all the list of activities that how blockchain is being done for a particular medication in a particular zone. Yeah, so this is more important and interesting, the clinical data management and data analytics. How the existing clinical data and lake of the data which is being there, say for example, in Punjab, one hospital is producing 10,000 patient data every day. Every month it is 3 lakh patient. Every year it is 40 lakhs patient. How can we take that data, integrate that data from different sources, and you analyze that data and you predict that data for the futuristic trends and activities, and giving the drugs and making sure, giving policies for the government that this is the way we are going in the right direction, this is the way we are going in the wrong direction, by using the data science models. How can you integrate the data? How can you standardize the data, quality the data, and predict the data analytics, which can be used for the futuristic purpose of the patients, is something which is very, very interesting story, and we need to discuss the stories more. Yeah, future is all about biomarkers, because the biomarkers are the one which can get into the analysis, the omics, data analysis, and also neural network modeling, and also the validation of biomarker, which you have received the really biomarker or it's a fake biomarker. Machine learning can prioritize the biomarker candidates, and further research and clinical trials and also data analysis can be done by using your AI-based tool. Yeah, the biggest problem with AI in pharma is the data privacy and security. So there is always the access control should be there, privacy should be there, and the data auditing should be there, and also the data should be cloud computing. So the biggest problem with AI is privacy and confidentiality and security of the patient data. It can be broken at any point in time. So we need to be very careful, because this data is very confidential, and as per HIPAA regulations, we need to carefully look into this privacy of the data with respect to this. Yeah, definitely real world data is useful. Now, already existing patients, nearly 10 lakh patients are suffering with diabetes, and they are taking metformin. But do you really think that metformin is giving an outcome for 10 lakh patients? Is don't know. How can you use the real world data and real world evidence and patient reported outcomes and give predictive analytics in future? Is something which is real world evidence, which is the data which you collected, you clean and structure it, and you give an insight and trends and technologies which can be used for the effectiveness of the patients and outcome of the patient for the futuristic activity, is real world evidence. Already patients are there, so many people, and real world data is jumping here and there. We just have to collect the data, survey our data, and then work on this data analysis. So this is something which is more important for us. And now it can be used in pharmacovigilance and adverse reactions monitoring. It can be used for signal detections, risk assessments, and it can identify any adverse event anywhere in the world, in a particular zone or gender or activities. You can directly identify, and you can look into this, that how the adverse events are being happening and how adverse events can be monitored by using the pharmacovigilance systems. So AI can be used in this also. And definitely precision medicine, not only the drug medications, but the nutritions, the health, and the walking and dietaries, and the genetics and predictives, lifestyle modification, everything. So it's not about just giving a patient a drug, but giving a patient a drug with nutrition, dietaries, and the lifestyle modification, and the social responsibilities and emotional and stress level, everything can be given at one place. That's your precision nutraceuticals and dietary supplements. You can engage the patients, you can screen the patients without going to the patient. By sitting here, I can identify how many patients are there by using sensor data, EHR data, EMR data, insurance data. Say, for example, I want to know how many people got accident. Simple example, one is hospital records, the one more is insurance record. The third one is FIR reports. So these three reports, if I take and I understand this, how many accidents have been happening, then I will come to know that how many patients I'll get for accident data. So something you can understand, the patient can be registered, patient can be identified virtually also. Yeah, so this is one more time. Earlier it was bench to patient. Now it is patient to bench. So I'll put it in this word, that earlier you go to a lab, you diagnose a disease, and then you go and identify a molecule and put it on the patient. But now you are identifying the patient, the rare and rarest diseases, and by using genomic analysis, and then going into lab and doing a research. So it's an opposite way. And now it's not bench to patient, it's patient to bench. So which is your reverse pharmacology. Your reverse pharmacology, based on which you can understand how this one can be done. Medical device monitoring and optimization. So you can put devices, medical devices, where algorithms can monitor your detection of potential issues in the body, malfunction in the body. Like Apple Watch can tell me that how my heart rate is increasing, my blood pressure increasing, and my activities are increasing, and all those things. It can predict the maintenance, and it can also give the usage optimization for particular activities. So now I think this is my last slide, and you can follow me on my YouTube channel and LinkedIn and Instagram and the other activities. But I'm open for questions. Any questions anyone have, you can ask me. I'm open for questions. I hope I have another 10 minutes of time.
Yes, sir. So I request to Dr. Kamya to further proceed with question queries.
Yeah. Any questions? I am open for questions.
Yes, sir.
So that we can have some discussions on the questions. I think Dr. Ravi Shankar sir also will be joining, I think, in short time.
Dr. Kamya, are you there? Hello. Sir, there are two queries.
Yes, sir. Yes, sir. I'm here, sir.
Okay, ma'am.
Thank you, sir, for such an informative question. We are having some online queries. Can I share the online queries with you, sir?
Yes. Please.
Sir, first online query is from Mitesh Sukhija. The person was asking, in medicinal chemistry, how should we use experimental learning methods or advanced teaching pedagogy?
Yeah, one more time, it depends if you go into funnel. It depends on how you can make use of medicinal chemistry. So how A plus B is giving a C reaction, you can do in a virtual modeling. You can explain in a virtual modeling, and then you can put it into the students where they can utilize it. This is a point number one. Point number two is we are using some of the data science models like Python programming for understanding the structural similarity of the drug and receptors, which can have bonding affinity. So it's a very deeper topic to be discussed, but this is how you have to do it. So more interesting, there are programming languages you can think about it, but you need to understand. See, AI is the ocean, but you should understand how you want to intervene with the AI is something which you have to understand very clearly.
Thank you, sir. There's another online query.
Yes.
From Mr. Amit Sharma. He wants to ask, what challenges do educators face when transitioning from traditional method to AI-driven teaching?
Yeah, see, the basic agenda is, are we ready really, is the question. So this session, I try to convince so much of things because it's 1.5 hour session, and 1 minute also, if you get deviated, then the whole session is going to a zero. If you get a phone or message also, your mind is diverting because it's a virtual learn. So how I can make 1.5 hour of a time engaging is something which you have to understand. And that's the biggest challenge. And one more time, we should get ready for adapting it. You have to get ready. You have to use more of LinkedIn and more of YouTube for learning and more of apps to learn. So there are some hurdles, definitely, how we can implement the technology. Excel presentations and technology like R and Python and statistics and data science models are more important for us.
Dr. Khadagraj.
Good morning, sir.
Good morning.
My question is there any AI technology for behavioral tracking in rats or mice in the preclinical study?
Yeah, there are models. There are AI models. Virtual models they have developed. And there are some systems which are being developed. If it is not developed, you can also develop. There are some models who have already put the steps. You see, the way you are thinking now is the right way. There are some models which are already put, and you can develop some more models which are not there in the existing market.
Okay, thank you, sir.
Thank you. More questions, sir? Any more questions?
Dr.
Ravi Shankar sir has joined, sir, by any chance, or he's joining?
Sir, he will join sharp at 11:30 a.m. So,
Okay.
Dr. M.D. Misbah.
Hello, good morning, sir. I'm Dr. Misbah, Associate Professor, ISF College of Pharmacy. I have the queries about the genomic analysis. Because we do genomic analysis, we predict any gene or biomarker from the patient data profile. It's good for the prediction for the any drug target to use for the any treatment like cancer treatment, anything. But we cannot be say it is 100% accuracy because the new drugs identified by prediction model, AI model, we need to be also be confirmed by some validation. We need to be confirmed by patient sample also. We need to be confirmed by some mice model or something. So we could not be say it's 100% accuracy or 100% efficacy for prediction by AI model. In India, this is a lack, there is a lack of the study about the pharmacogenomics in the country like US, Australia, which is developed countries. They already have the developed about the genomic analysis and drug predictive. But in India, it is a less sufficient. So we need to be more study or more project. We need to be study about pharmacogenomics study. Thank you.
Yeah, I understood your question. See, the point here is nothing can be 100% sure. Even my session cannot be 100% sure for all the 200 people. But what is a percentage of that reality which can be taken is something which we have to understand. There is always a validity and invalidity, and 100% it cannot be perfect. That's a point number one. But at least it's 60%, it is okay. We are covering that 60% of time without wasting that another 60% of molecules. That's a point number one. Point number two, definitely I accept that the way the technology has grown for the other countries, India might not have grown that way and pace, but nothing is late and we need to start immediately. So the sessions which I have taken might be eye-opener for everyone, and faculties might get connected to me later on also. So I'm open for further discussions that how we can together take India as a health capital or India as the best country in the world. So that's something which we can always open up our eyes and look at it. But definitely, yes, it cannot be 100% valid and 100% truth, but the amount of percentage can be less. So that's one thing. And the other thing, yeah, other countries have developed a bit, yes, the way their research is being done. Yes. Perfect. Any other questions, sir? Or
So there is one last query in
Yes.
online YouTube chat box, and the query is, what is the precision level of AI? Does it replaces human service in clinical practice?
Answer is no, because every patient is different. Every patient is different. I might sleep only for 7 hours, 40 minutes, but only for today. But for another 10 days, I might sleep only for 6 hours. I might walk for 10 kilometers today, but another 9 days, I might walk only for 6 kilometers. I might have taken 1 bottle of alcohol yesterday. I might not have taken 10 days alcohol. I might be stressful because my dog died, but I'll be happy that I'm watching movie daily another 10 days. Everything depends on individual patient to patient. The clinical outcomes cannot be automated by AI, but we can take proposals from AI. With the use of AI, we can spend more time on data analysis and data science and data analytics. And how can we grow it? We can do it definitely.
I request to Dr. Kamya proceed further.
Dr. Kalicharan, Manji Kalicharan also have one question.
Okay, sir. Yes, Dr. Kalicharan.
Good morning, sir. I have one question in relation to virtual screening in small molecules. So there is an option to validate the docking protocol. So while using AI-based virtual screening or generative AI-based model, so is there anything to validate our results from that area?
Yes. Answer is yes. See, I'll tell you something here. The AI results might not be 100% perfect, and validation of AI might not be 100% perfect. I'm accepting it. And even ChatGPT will give an option at the end saying that actual result might vary. You just have to go and verify the other resources. That's the beauty it is being given. AI can propose us, but you can't just take the AI virtual screening and docking methods and say that AI has given, you take it. It has to be a human intervention which has to confirm the validation of the results, or else it cannot be done.
Okay. Thank you. Thank you, sir.
Thank you.
Dr. Kamya.
Thank you, sir, for such an informative session. Now, I would like to invite our worthy director, Dr. G.D. Gupta, sir, to conclude the session. Sir.
Thank you very much, Dr. Kamiyaji. So first of all, I am thankful today's speaker. He is the very, very dedicated experience, and he delivered the lecture. This is recently, you can say, very important for each and everybody for the revolution of the education. So ISF College of Pharmacy, thankful Mr. C.S. Mujabuddin, and he included number of the good things. And one thing is the specify that teacher can be removed. What the teacher cannot be removed, this is important, but the content and the material what we are the delivering, it may be removed or it may be changed. So we need to the change and tune with the time and temperament. And obviously All India Council for Science and Technology as well as number of the professional body, if you want to the tune with the technology as well as the education, so this is the everybody is the required. We need to the work on the technology and tune with the system. So Dr. C.S. Mujibuddin consider number of the parameters, each and every pro and cons, as well as he is the given number of the good thing. This is related to the drug discovery as well as the development. So obviously number of the models available, but it is not essential that all models are sufficient. So this is the capacity of the teacher. You can develop and provide the information whatever is required for the society. And as I know that recently one of the university in the Lucknow is especially based on the AI is the going to be the start in 2025 and 26. Each and every parameter it will be the based on the AI. And obviously one thing is the given. One word is the zero paper policy. Obviously, it is very, very important. So if any teacher, even it is not required that you can implement the whole of the process of the AI, but one teacher and one lecture, you can accept it and implement it in your lecture. There is the no zero paper policy, and you feel that it will be the helpful to the student or not. So obviously, each and every technology have their own values, own advantage, as well as the own disadvantage. It is depend upon the person to person how you can take it. So thank you very much, sir, and you have given lot of information in technical manner, scientific manner, and obviously the young teachers, they can be do so many good things and tune with the artificial intelligence application in education system. So from core of my heart, I thankful to the whole of the team of this FDP program, provide us such a opportunity to interact such types of the very, very learned and dedicated person. I am thankful to the especially APTI Secretary Saab, Raman Dangji, for provide this opportunity and give your name, sir, because the first time I can connect to you. And obviously you're always welcome at ISF College of Pharmacy. And I want to start one of the program, if you can give the permission with your collaboration, one of the certificate program in the ISF College of Pharmacy, so we can collaborate it and our team will connect to you so that it will be helpful, especially in the pharmacy. I know that artificial intelligence, their application implemented in the number of the computer program, engineering program, number of the branches it can be applicable. What yes, the application of the AI is very less in the pharmaceutical education. I hope coming days it will be the grow very fast. So thank you very much.
Thank you, sir, and thank you so much for giving nice words, sir. And I'm very fortunate that I have received the award on the hands of G.D. Gupta, sir, in the Pharma Lok. I was in Delhi for one of the workshop in Jamia Hamdard, and I took one award of Pharma Lok in this year. Very, very thankful, sir, for your support. And we are happy that we can take some group of students from ISF College for some research projects for surveillance studies and clinical data science and AI-based models for the benefit of the patients. We can do some workshops, certificate programs, and we can have MOUs, and we can further see that how we can get the students more and more active, sir.
Thank you very much, sir. Thank you.
Thank you, sir. I think before we get Ravi Shankar, sir, I think Ravi Shankar, sir, I just called and sir said that he will join in another 4-5 minutes.
Yes, sir.
Ravi Shankar, sir, is very, very hardworking and very, very busiest person who has accepted at the last minute for our event. He's traveling to US today afternoon, but because we have invited and he has seen that ISF College has this FDP program. He has postponed his travel for US, and he is traveling tonight for US. And he is a cardiovascular surgeon who has interest in his Ayurveda research, and he has done so much of research and endless of awards and endless of activities he has done. And we are very, very happy that Ravi Shankar sir is joining for us to deliberate a session. He has done a wonderful research and he has developed his own devices. So a device which can be connected on the wrist and will give how your organs are working in the body. So he has really implemented AI models, and he always supports me for any workshops and certificate programs. Wherever we do, he comes and he delivers his sessions. Though he is very busy, he understands how we are doing the activities in terms of Clinosol by providing a lot of hands-on workshop and hands-on trainings and all those activities we are doing it. So he always supports us, and he has developed a very good models with Vata, Pitta and Kapha models. In animals also he has put, and in human beings also he has put. He has more than 100 patents on his names, and he's one of the best cardiovascular surgeon who has worked in US and also Canada and also in Russia. He is starting his own Ayurvedic medical college in Europe. He's a speaker in UN also, United Nations, and he's a well-known personality where every IIT and IISc wants his sessions to be delivered. So we are very, very fortunate that he is joining us. I hope he will be joining in a minute if I'm not wrong. So his part of research was more towards end-stage patients. Like say, for example, a patient cannot be treated for any system. Ayurveda, allopathy and homeopathy, any system cannot treat it. He says that I can treat that patient, and he makes sure that he treats that patient. His case reports, his patients who got treated, the patients who came into his hospital without walking on a wheelchair, and then the patient is returning with walking with his Ayurveda and integrated with allopathy, Ayurveda and also herbal medication. I think he is the one of the best person to listen to us that how the medical research is developed. And his vision is very, very clear that how India can become the health capital of the world. He has started his 100-bedded ICU hospital in Hyderabad very recently, and he is doing a lot of research at end-stage cancer and end-stage, any patient end-stage who cannot be treated. He gives more of treatment options with the help of Vata, Pitta and Kapha with Ayurveda and allopathy. So he's one of the best person to listen. I hope he is joined. And after this session also, we have one of the person who will be joining from Delhi. His name is Dr. Sridhar. I'm very thankful for Dr. Ravi Shankar and Dr. Sridhar who has accepted at the last minute because these people support me on all kinds of workshops and conferences and all those things. So he's joining from Delhi. He's doing his AI in Pharma from Boston University, and he's doing his research work in Delhi. Now he's in India. So he has worked as Senior Vice President in Hetero also. He'll be joining at 12:30. So this session also will be very, very interesting and very, very engaging session wherein we can learn more things from Dr. Sridhar also. And Dr. Ravi Shankar, I think definitely he is the one of the best person to be listened. And he's always my guru. And the activities, the engagements which we are trying to do for the next level and next students, any people can call us and we will be happy to come and give session. But the most important agenda is people like Ravi Shankar sir has grown us in this level that we can put a lot of knowledge to the students. Sir, let me know if Ravi Shankar sir has joined, sir, so that we
Not
can.
yet.
Yeah. So I think sir said that he will be joining in a couple of minutes.
So one person, Dr. Divya, is corresponding from his side. So I think
Yeah,
he will join
people
in
can
5 minutes.
ask questions if they have anything, sir.
So almost all queries have been resolved by you in a very efficient manner. So we move to the next session that will be coordinated by Dr. Kamya also. So we wait for that.
Yeah. So if any other questions are there, we can have discussion, or else we'll wait for a minute where sir joins us for the session.
Yes, sir. We will wait that sir will continue.
Thank you, sir. Now we are waiting for Dr. Ravi Shankar to join the session. It's been a couple of minutes only.
AI scientist and Python programmer and Ayurveda scientist. He has done so much of research for end-stage cancer and end-stage patients. He has saved so many lives, sir, and we are very happy that he has accepted after I saying this FDP program. So he postponed his travel also for our session. So we are fortunate to have him today. So he said he's joining in a minute.
Excellent, sir. Excellent. And we are also very, very impressed with your lecture. And obviously, you have given the reference, so it will be also very fruitful to the each and everyone. Because I know that if we accept or opted any types of the technology, it will always created some of the problem and pro and cons. Obviously, in the initial level, when we have started the computer technology in the pharma, then most probably the obviously the peoples is the don't want to accept it the easily. But nowadays everybody is the technocrat, and from the starting they have the well known with the focus with the computers, computer applications, so it will be the easy and helpful to the each and everyone. Number of the question is the find given asked by the our faculty members as well as the delegates, those who are the registered in this APTI. Okay, if this will be the replace the teacher, not replace the teacher, or there is the no any personal touch. So obviously time is the change. And if you want to your own respect, one question is also given one word by the C.H. Mujavuddin ji. One word is the respect. If you want to the respect, you can respect your work, you can respect your duty, you can respect the technology, you can respect the homework so that you can achieve the goal. So thank you very
Dr.
much.
Ravi has been joined.
Yes, so welcome, sir. Welcome at ISF College of Pharmacy. Myself, Dr. G.D. Gupta. So, sir, welcome.
Sir, good morning, sir.
Good morning. Good morning,
Yes,
sir.
Good morning, sir.
Good morning. Thank you.
Good
Thank
morning,
you.
sir.
Thank you.
For the next online session, our speaker is Dr. Ravi Shankar Polisetti, CEO and Director R&D, Sai Ganga Panekia Private Limited, Research Consultant, Intellectual Property, Johns Hopkins University, Baltimore, Maryland, US. Dr. Ravi Shankar is an experienced researcher with a demonstrated history of working in translational research. He is skilled in research and development, clinical research, animal models, Python, AI, ML, Watson analytics, medical devices, and a scholar well versed in Sanskrit scriptures and a member of prominent global cardiac surgery associations, including IAHPC, PCSI, and many more, and the Russian Avia Cosmic Medical Association. He is strong research professional and a cardiovascular surgeon, and for the first time demonstrated subtotal regeneration of the heart tissue in post-MI animal models. He holds several patent pending technologies in healthcare and AI/ML fields, 17 patents filed and another 41 are undergoing search, and is recognized with the several national and international awards, including the Lead India Bharat Ratna memento, among others. He is editor of various scientific journals, advisor to various biotech, clinical research, and pharma companies. His topic for the session is AI and ML and the future of drug research. Now, I would like to invite Dr. Ravi Shankar for his today's FDP lecture. Sir, over to you.
First of all, good morning, everybody. And my Google Meet was muted. So when you all wished me, I wished back, but no one could hear me because I was muted. Dr. Gupta ji, thank you very much. Good morning, everyone, again. And Dr. Kame, thank you very much for the introduction.
You,
and I cannot enough thank Dr. Mujib for introducing me to this wonderful group. I'm so happy to be connected with you all here. I just made a quick presentation on, because my topic, as Mujib said, कि सर आप AI ML में भी मतलब AI ML में माहिर हैं, तो why don't you explain us the concepts and the future directions of AI and ML in pharmaceutical industry and and its application in the yeah. Can you all see my presentation, please?
Yes, sir.
Okay, cool, super. Yeah. So that's what Mujib ji mentioned. So I said, okay, let me just prepare this for you. 1 second. Is this the one? One second, please. So I'll quickly, walk you through some important applications of AI and ML in today's world. And of course, I'll touch base on the current applications in the pharma industry and also on.
इतनी देर से मैं ये परेशान हो रहा था कि स्क्रीन
of AI and ML, and when it comes from India, I think we can actually add a bigger leaf to the entire story of AI and ML, and possibly, if we are working together, we'll help or will be a part of, that history where, we all could be and would be considered as, pioneers of making India the health capital of the world. And I really tell you, friends, we really have that potential. So let me just open the PowerPoint, please. 1 sec. Couldn't share it? Oh, yeah, this is about share. Look at what yeah, perfect. Yeah. Finally. So, I'm a avid Zoom user, so so it's Google Meet is slightly newer to me.
Visible to all of us.
Yeah, it's visible.
Visible to
Okay,
all
great.
of us.
Excellent. All right. So let me just, I wanted to give you a quick insights into the application of AI and ML in pharma industry, in drug discovery, and the so-called polyscientific advanced medicine. We'll just walk you through this. So what I would definitely, want my audience, most of them, if not all of them, are from the pharma industry, right? Either professors or faculty in the pharma colleges, and or students who are who definitely want to become or make their name in the pharma pharma world. That's what I'm assuming, and that's so the role of AI and ML, friends, machine learning and cell analysis. And there is something called as in the drug discovery process. Machine learning algorithms have immense benefit in analyzing in the complex data sets. What happens is, for example, when you talk about a process in drug discovery, what you have to do is what we usually do is we just add a compound known or unknown compound, usually unknown compound, because we want to see their effects on the cells. So what do we do with that? Once we have this compound, you take in vitro or in vivo. I mean, in vivo we'll do it later, but then now we have, with the help of these cell culture technologies, you can always take a cellular assay, put this compound in the cell, and traditionally you got to look at these cells through your microscopes. There are advanced microscopes right now, but then with the advantage with the ML, machine learning, or AI algorithms, is that you put this compounds in the cell culture, and then you take a picture. I mean, I'm just trying to simplify it. You take a picture of the normal cells, which normal healthy cells, and compare the pictures that are taken from under the influence of different compounds. So what happens? The algorithm automatically checks if there is any division of the nucleus, or if the cells are divided, or you have a botched nucleus or a micronucleus, or there's a specific machine learning algorithm called as computer vision. So the computer vision uses a specific statistical method called as principal component analysis, and it takes a picture of the cells and then and compares it with the with the normal cell image, and you essentially end up with profiling several hundreds of these images in really a trice. That's like going to really help you understand data very rapidly, and then you can make informed decisions very quickly, and then research will be literally streamlined, and then you're essentially have a lot of advantages, and because of its rapid data analysis over traditional methods. Similarly, in visual inspection in drug production, what happens is in a drug production, most of these drug production or manufacturing companies. I was once listening to the lecture of a director of one of I think either Novartis or Pfizer, and Dr. Vijay, if I remember his name right. Now he was explaining how pharmaceutical companies lose about 3% at least of their revenues. I mean, that's huge. 3% of their revenues, just because sometimes visual inspection of the drug production is not sometimes 80%. That's the number that he gives. That 80% of visual inspection leads to wrongly estimating, wrongly analyzing a right product. You get false positives, and then 80% of these products are rejected. So that amounts to more than 3%. 3% is huge. So instead, again, applying the same computer vision technologies, you essentially take pictures of those. I mean, it is called as a labeled data, which means your data or the pictures that you're taking of the production unit or the production tablets, which you consider as normal and absolutely fine, they have to be compared. I mean, you essentially generate a data set with all the normal tablets, and you train the algorithm. This is called a supervised learning. So you tell the algorithm, this is normal, so you register that. And then there are millions of nodes inside them. There's something called as a convoluted neural network. That's part of the neural networks in this category called the supervised learning. And the algorithm automatically applies several statistical methods. You can't even imagine. It really tries, applies all these algorithms, learns from you, from the images that you provided, and the images that you say are marked as normal, and then immediately creates that algorithm. And this is called as machine learning. So the machine is learning, and then that is converted into an artificial intelligence program or a algorithm, that when it is given a challenge of now that you have trained the model, then you essentially deploy it in a production unit, and it automatically detects whether there is a problem or not, and with which Pfizer says that it saved at least $70 billion. That's the advantage of this machine learning, and it all happens with in really 100x times. A human error definitely is a big problem, but then there's automated systems definitely could revolutionize the way we do drug inspection. So I'm just giving you certain applications of how AI and ML are used in enhancing inspection, and also integrating that for improved efficacy, efficiency, and compliance, friends. So what we did was, there's all this AI and ML, we try to understand how they could be applied in the field of medicine. So now we know that, we had been doing this research on correlating Ayurveda with the modern science. And we were possibly the only ones in the whole world to create models of Vata, Pitta, and Kapha in experimental animals, and then correlated that with the electrophysiology, the cytokine studies, and then we generated a lot of data. So I was just talking about data integrity. That's what we did, and then arrived at this so-called polyscientific advanced medicine. So right now, where all is AI used is, if you look at, okay, how the ancient science understood the modern sciences, I mean, the physiological processes is that they classified everything that you find in the body were pretty much classified into three broad categories: the Vata category, the Pitta, and the Kapha category. So it is called as the lines of separation of data. And why are they important? I'll come to that very quickly, friends. But this classification has helped understand which metabolic or physiological pathway predominates in which individual. And then, kind of, it gives a kind of data pattern in the human body. And then, this is already included in Ayurveda, not the routine Ayurveda, friends. You don't confuse with what is being practiced as Ayurveda today and what is originally meant to be Ayurveda. They're completely different. Being a cardiac surgeon, I can tell you that, that's not how our ancient scientists have designed. But currently, Gen AI or AI is being used. There are several models that have come, new models. Models are being created literally, I mean, generated literally on a day-to-day basis. It's very, very fast transforming. And why is it important, friends? Today, see, if you look at the data available as on, there was an article published in NEJM in 2019, 2016, in which, or even earlier, 2015, sorry, in which, doctors were the leading cause of death, actually, according to a certain ranking in the past. We as doctors were rated at 3. I mean, the number 1 cause of death was heart disease. Number 2 were COPDs. At number 3, doctors used to come. Now, thanks to the WhatsApp University professors and YouTube lecturers, friends, we are pushed to the level 6, friends, because the WhatsApp University professors are thinking, and also they confidently tell you stuff that they don't know. And they post their videos just for likes, and then people blindly are following. And I tell you, we routinely encounter these patients in our hospitals and clinics, and we see that these are all people, patients who are who were trained and advised by WhatsApp University professors, and they pushed us. The 3rd spot, the 4th spot, 5th spot are taken by these WhatsApp University professors and YouTube lecturers, friends. And that's very, very dangerous. And that is like, we are pushed to number 6 today. But, jokes apart, friends, but is AI helping the modern medicine? Is it useful, friends? I would say yes. There are several techniques applied today. Algorithms applied in revolutionizing medical imaging, and then deep learning and diagnostics. And there's several real-world examples of AI success in medical imaging, friends. So, for example, right now, I'll just quickly breeze through where all AI is used. AI is used for early detection of a disease and predictive analysis and overcoming simplification, the streamlining of the healthcare processes. If you have a EMR, then administrative burden is off from you, because you have the AI taking care of streamlining your healthcare processes, improving patient care. And then for example, retinal imaging. I never even thought, I never even guessed that the retina, the image of the retina would give you such deep insights applying AI, and you could precisely predict the heart's calcium score. The heart's calcium score cannot be predicted unless and until you do in the current arena. The calcium score of the heart in ischemic heart disease is predicted only by a certain angiography study called as angiography. But now with the retinal imaging and you could come up with AI that will help you essentially detect your calcium score, and then it helps in prevention of several heart diseases. Retinal imaging also is used in Alzheimer's disease, in detecting Alzheimer's disease, in diagnosing Parkinson's disease. It also gives a neurological insight into helping retinal image, just giving so much of data, friends. There are several studies published. You can always find them on the internet, like Jove Wise studies in Nature on a foundational model for disease detection from retinal images. It's an amazing breakthrough, friends. And Gen AI is again taking AI to the next level, friends. For example, ECG analysis. Can you imagine that a ECG could give you your right ventricular systolic pressure? We would never have imagined that a ECG. From ECG usually used only for detecting your rate, rhythm, and axis of the heart, which give certain insights into how the heart is functioning. That is like a preliminary study. Of course, you can detect ischemias and then order for the studies like 2D echo and the TMT. But then ECG gives you the cardiac functions, ejection fraction could be detected with the help of ECG. That is the power of AI, friends. Now, heart pressure sensing, ECG's capability into detecting of the heart pressure, the LV pressure, the RV, the left ventricle and the right ventricle pressures are right now used. These are all already published studies. They will soon take over your lab, investigation, or your doctor's burden to detect a disease. So ECG is like, this is all is possible with the help of AI and ML, friends. Anemia, hemoglobin levels, diabetes and pre-diabetes conditions are now being detected with the help of AI, friends. With the help of just ECGs. Chest X-rays, they're even predicting the race. They're even able to come up with ethical considerations, and that's really going to be a dream come true for a doctor, because it's the investigations itself usually would take anywhere between 7 to 10 days. When you encounter a patient for the first time, it takes a lot of time. So here with this, there are several methods of AI. I don't want to go deep into the GELU or the fully connected layers or the transformers, and I don't want to talk about it because that's going to be too much for this discussion. But suffice to say that there are so many models that have been arrived at in the last 7 years. BERT and transformers and NLPs. NLP is natural language, and of course that's what is used by ChatGPT and other Gen AI platforms. So friends, so the basis is your the key is coming again from your fundamental understanding. So neural networks are actually inspired from neurophysiology. So the scientists have understood what is happening at a receptor level, how the information is being processed by the body, by the brain, how the information is sensed at the side of receptors, then transmitted through the nerves, and then what does the brain do? What are the centers, the cortical centers, the subcortical centers, then the limbic system, the substantia rubra, the substantia nigra. People have analyzed this already. Mark Turin, I suppose, would be the first who would have started this neural network program. But then it evolved over the last 70 years, friends. And when I read these neural networks, the architecture of the neural networks, I really I'm amazed to see how the fundamental architecture of a neural network is actually inspired from neurophysiology, friends. So we'll come to that a little later. We don't want to talk about this. Okay, I'll skip this, please, because this was specifically for AI and ML engineers. Yeah. So what we did, friends, was we tried to understand the foundations of Ayurvedic theory. So what the ancient scientists were saying, first of all, why do we need them? We'll come to that a little later. But what has happened is India is a diabetic capital of the world. We all know that, right? About 18 crore patients pretty much, suffering from diabetes. And added to that, there are 7 crore who are categorized as pre-diabetic. But the problem is, according to the American Endocrinologists Association, you are a diabetic when your HbA1c, we all know what is an HbA1c, right? The 3 months average in a diabetic patient, according to American Association of Endocrinology, they're saying it should not be beyond 5.5. That's an average of 120 pounds. But then the American Diabetic Association is saying, no, we are not going to be too rigid. It should be below 6.5. That's their target. That they're trying to maintain it below 6.5. They say that anything above 5.7 is pre-diabetic. That's the American Diabetic Association's, pretty much say. And then comes the American College of Physicians advice that came in the year 2019, when they retro-analyzed more than 3 million Americans, and they did, by which time they finished a specific trial called as a prospective trial called as ACCORD. The ACCORD trial has given them such deeper insights, friends, and based on which they would say they are actually, this is already gone into the guidelines on the ACP's website, American College of Physicians online website, according to which they're saying your HbA1c should be between 7 and 8 and never below 6.5. Remember this, never below 6.5. Because and they are at loggerheads with the American Endocrinologists Association and also the American Diabetic Association. So because the Diabetic Association is saying your HbA1c should be below 6.5, the American Association is saying, College of Physicians is saying it should never be below 6.5, because according to the study ACCORD, they figured that especially when a patient has concomitant heart disease along with diabetes, such people, 1 in 3 in that trial died of a heart attack when they were subjected to intense glucose lowering. When they pushed their HbA1c levels below 6.5, 1 in 3, which means 33% of these patient population died, and they had to abort the trial. And hence they're saying that your HbA1c should be between 7 and 8 and never below 6.5. And in some instances, based on a certain study called as a VADT, Veterans Affairs Diabetic Association, they're saying that your HbA1c should be around 8.5, because these veterans, again supported by, or these guidelines come from American College of Veterans Association. American Association of Veterans. They are saying that these veterans were doing good between 7.5 and 8.5, and even 9.5 was good in some individuals, because in those patients, in some of these, or at least 30% of these patients, when they pushed their HbA1c levels through intense glucose lowering strategies, when they pushed below 7, they ended up with hypoglycemia. What is diabetes for an American endocrinologist or a diabetologist is being considered as not only norm but an essential HbA1c level for a veteran. How do you beat this? The problem is, according to who are our 25 crores of Indians being categorized as diabetic people? Of course, we do have ICMR. By the way, okay, I'm not discrediting any organization, friends. I have immense respect, because they all have come up with these numbers with the help of data. See, in all, I pulled up all the 750 clinical trials, not just studies, the clinical trials and retro studies, of course, analyzing the metadata that they had in the last 45 to 50 years. They pulled up all that, and then they analyzed about 17 million Americans, 1.7 crore Americans. Data was analyzed, and then based on that data, all these institutions are giving their recommendations. And what is working for the American Veterans Affairs Diabetic Association is not working for the American Endocrinologists Association. So ICMR definitely has done some work. It has pulled up about 16 clinical trials in different places, but then the size of the population, the size of the sample that they analyze is only 2,000 patients. They did 16 studies with 2,000 patients, and they are saying no, it should be 6.5, and it should never go beyond 6.5 is what ICMR is saying. But for for the size of the India's population, for 1.5 billion people, your data sample of 2000 is extremely low. You cannot even make any meaningful conclusions according to me. And the Americans, on the other hand, they have come up with data from 17 million Americans. So that's what I wanted to say. We have done 16 studies, they have done 750 studies. So you look at the data that they generated. But then what do we do now? How do we treat our patients? Because we see routinely, as a clinician, as a cardiologist, I keep seeing patients, really, some patients do exceptionally well with 8.59, but some patients end up with diabetic retinopathy, diabetic nephropathy, and several diabetic neuropathy, peripheral neuropathy at 6.5 to between 6.1 and 7 itself. So this is the dilemma, friends, and the question that I would ask is, can we offer any deeper insight into what is happening now, and can that be possibly derived from our own ancient wisdom, friends? Because as a last 32 years, this is what I had been doing. I was trying to analyze what is given by the Americans, what is hypertension. Again, as per the Americans, is not hypertension as per the European, because the American Heart Association says that your normal age blood pressure should be below 110 by 70. Anything above 110 by 70 should be treated. And this is not driven by pharmaceutical, friends, because unfortunately, these WhatsApp university professors have ruined the intellectual capability of people. And I would completely disagree with them. There might be some pharma lobby. Of course, there are lobbies for several parties, but then the pharma company is extremely regulated. And companies are regulated. There are serious organizations that are overseeing the functioning of these different companies. You have the US FDA, you have the CSIR, I mean, not just the CSIR, the CDSCO in India. These FDAs are very strict with data, friends. You cannot just come up with numbers from the thin air. So what we did was to understand this problem, we try to approach the Ayurvedic way. So what Ayurveda is actually talking about is that saying, okay, by the way, Ayurveda, is it a primitive science? I wouldn't agree, because I've done a lot of research on that. But is it advanced? I would say yes. But how advanced is it? Can it be used as is mentioned in this text? I would again disagree. No, you have to do a translational research of Ayurvedic concepts, understand what the ancient scientists were talking, and then be able to apply this for the modern. And there's several problems here too. You cannot just straight away pull up, the demographic analysis given by Vagbhata Maharshi in Ashtanga Hridaya and then apply it to the demographic of today's population, because the populations are seriously mixed up, and they have different VPK fingerprints. So what is VPK, and why is it so important? Does VPK lead to a very advanced AI algorithm? Is what is going to be my line of thought and lecture in the next 9 to 15 minutes, friends. So, and by the way, yeah, are these numbers? Does Ayurveda consider numbers? Yes, there is a certain concept called as Anguli Pramana and Anjana Pramana, the qualification analysis and also quantification analysis. So both quantification and quality determination were also matters of subject of Ayurvedic practitioners of the ancient times. So knowing the Vata, knowing the quantities alone won't help understand health, and hence Ayurveda postulates very strongly that there are three agents in the body called as the Vata, the Pitta, and the Kapha. And when these three agents are in equal proportion in all of the 7 tissues and 42 organs, then the body sets itself into a self-healing mode and cures itself. That's what Ayurveda says. So there's a specific, verse in the Ayurveda. It says, Samadosha, Samagnischa, Samadhatu Malakriya, Prasanna Atma Indriya Manaha Swastya Ityabhidiyate. So this is the original shloka mentioned in Ayurveda for defining what is health. So it says, when these three agents, the three doshas, samadosha, I'd say. So the 3 agents are in equal proportion in all of the 7 tissues and 42 organs. Then normal metabolic physiological processes happen in the body. The excretory wastes are eliminated from the body. Then a person ends up with prasanna atma, indriya, and mana, which means they ends up with physical health, mental health, emotional, social, and spiritual health. So, and then the 7 tissues, again, they have their own classification, and there are pancha koshas. I mean, I'm not going to go deep into all of these concepts, friends, but then where did we, is data science applicable to Ayurveda is what we're going to discuss. So now I just mentioned, the World Health Organization also mentions health as a complete physical, mental, and social well-being, not merely the absence of a disease. That's exactly what modern science says. But we already talked about the different standards mentioned in the modern science, and how this is not helping, because what is helping a certain contingent of patients is not helping a different contingent. It's actually contraindicated in a different set of patients. Again, the same thing. If you talk about the cholesterol, friends, there are definitely benefits of lowering LDL, the bad cholesterol, in some individuals. At least 49% of patients have got benefited, but the 51% of the patients who are put on cholesterol, they suffered from cholesterol, the statins, which is HMG CoA reductase inhibitors, did good to 49% of people, and in rest of the 51, they actually, out of which there's a certain population who ended up with a new form of heart failure. The very purpose of cholesterol, the statin drugs, is to help the heart. It's a cardioprotective agent, but it has caused a new form of heart failure. I'm not saying this is a Norwegian's Hunt study that is actually saying not only this, it led to rhabdomyolysis, it led to AKI, acute kidney injury in some individuals, it led to liver failure in several individuals, it led to early onset of senile dementia. That's what the study says. It's an elaborate 92,000-page report, friends. And they have done a meta-analysis of more than 95,000 patients. And based on that data, they're coming up with this, numbers. And again, people hold some conspiracy theories against this cholesterol. Nothing of that sort, friends. It's just the data. The Westerners are looking at data, and I see how they are right too. I'll tell you towards the end of this next 15 minutes, you'll figure that they were actually doing it right. But there's something more that we could contribute to arrive at proper data analysis, and then help them understand their own data. And again, Dietary Guidelines Advisory Committee, the DGAC report, it says there's no correlation between your cholesterol from food and cholesterol, and the cholesterol, and the heart disease. That's what was published, not just published. This is a presentation made by Dr. Alice Lichtenstein in the year 2014, 11th of December, to NIH, National Institute of Health, and also the US FDA. So friends, how do we, the problem is now there is enough data which mentions also that your low levels of LDL is also causing heart disease. High levels of HDL in certain Caucasians, Iranians, Iraqis, Northwestern Indians, the Baluch Republic people from this Caucasian belt, they are essentially suffering. They would suffer a heart attack if their HDL levels are high. Of course, there's a certain there's a certain genomic expression that is correlated to that. But then, how will you beat the fact that even high HDL in these populations is actually causing heart attack? We already know that low HDL is causing heart attacks. High LDLs is also causing heart attacks. So the problem is when a patient comes to you, how do you actually treat the patient? Is the biggest problem. Whether you're going to lower cholesterol, whether you're going to give him a statin or not. But the American Heart Association doesn't modify its recommendations yet. It's not that they don't want to, but they don't have enough data yet. But then the physicians definitely are confused. The cardiologists definitely are confused as to in whom the statins are helping and in whom are they not helping. So there are several problems, friends. So a quick summary of how the modern medicine, where does it excel, and what are the gray areas, friends? So what we did was to try and test any hypothesis, friends. You need data from live patients. So we had pulled up that category of patients who are not, who are essentially excluded from the treatment methodologies or from the guidelines, like end-stage heart diseases or end-stage kidney disease, end-stage. Even when you encounter patients with heart failure, when the EF is less than 30, or end-stage cancers, 4A stage cancers, when the patient is left with just about 3 to 6 months of his life. Those are the areas where still a lot of contribution from the modern medicine, or in the current form of medicine, we could essentially help that category of patients, and hence we try to work with these patients, friends. And as I mentioned, there are certain gaps that we could potentially fill from the ancient wisdom of Ayurveda. That's what was. But we don't want to do it rhetorically. We want to do, essentially test it, understand the mechanisms or the concepts mentioned in Ayurveda, and then approach in a scientific way by generating data, friends. So I already mentioned that when these three agents of Vata, Pitta, and the Kapha are in a state of equal proportion in all of the 7 tissues and 42 organs, if you translate that into the language of modern science, when these three agents are in equal proportion in at every level of biological interaction in the body, the genomic level, genes lead to the production of proteins, then the proteins trigger certain enzymatic pathways, certain receptors, all guide and these receptors in turn guide the subcellular pathways, then the cellular pathways, and the tissue, organ, and organism level of interactions at every level. If you a biological interaction, if you're able to balance these three agents, then you essentially end up with health, is what Ayurveda is saying. This is all hypothesis, friends. This is all what is mentioned there. So if you do this, then you end up with physical, emotional, social, mental, and spiritual well-being. But the fundamental problem was that Vata, the Pitta, and the Kapha are characterized by some organoleptic properties, by their certain properties. These are called as the lakshanas in Ayurveda, which pretty much as of yet don't make much sense. What is cold? We don't know what is cold really, right? How does it affect the body physiology? We really, as of yet, if say you take 1.5 pH of HCl, whether it is cold or hot, it doesn't matter. It exerts it has its effects, right? And these effects are not much different. That's what the modern medicine, at least the modern chemistry says. But then Ayurveda, because we're trying to understand the basics of Ayurveda here, what we try to analyze or understand is whether, what is mentioned in this text is first. That's what we were trying to do a literature review, and figured that Vata is characterized by 7 properties, Pitta by another 7, and Kapha by another 7. And they pretty much, like if you look at the translation of Hima here for Kapha, it's translated as cold. And again, for the Vata is characterized by a certain property called Shitala, which again is translated as cold. But then here, from the words itself, we could figure that there is something seriously wrong, because Shita is translated as cold and Hima is also translated as cold, but there are 2 different words. And in Sanskrit is a very advanced language, friends. It's not just straightforward language. It's got if Hindi, for example, the English, for example, has about 982,000 grammar principles. And Hindi has about 1700 grammar principles. Vyakarana, it's called as the Hindi Vyakarana. And the Telugu Vyakarana, or it's called the Chando Vyakarana, has about 1600 principles, friends. Sanskritam has about 200,000 grammar principles. 200,000. They're classified into 9 different libraries. These libraries are called as in Sanskritam, they're called as a Nava Vyakarana Bhasha. That's why the language is called as a Nava Vyakarana. It's got a 9 different set of rules guiding the way you should understand a certain mantra. So, and again, the Vyakarana Rishis, or the grammarians, the scholars of grammars, they mentioned that Ekamatra Lagavante Putrotsaham Manente Vayakarana. Even if you reduce your communication by just 1 matra, the Vyakarana Rishis will be so happy, as if a great legacy is born for them. Great offspring is born to them. That's how happy they're going to be. That's what Jagat Guru Adi Shankaracharya mentions. And then here, and again, if you look at the language, I mean, the translation of the word Shita, it's mentioned as cold, but then the word Hima also is translated as cold, but then these two words of Shita and Hima are completely different, isn't it? Their matras are different. And then if the matra is different, the meanings got to be different. That's how I started, further reading these original Sanskrit scriptures, understanding Sanskrit. As I already mentioned, there are 9 different Vyakaranas guiding the language of Sanskrit and the way you understand these rules. So these are the Indram, Aindram, Sandram, Shakalam, Shakadayanam, Krashakursram, Kaumarakam, Paniniyam, and Sarasatam, in all 9 different grammars. And what you have to do is each matra should be understood from each of these 9 different Sanskrit grammar sets. So when you apply one principle from each of these 9 grammars, you end up with 9 different meanings for each matra. If a word has 2 matras, then you end up with 9 to the power of 2 meanings from. That's the effective way, very, very effective way of communication. We all are proud of Sanskrit, but do we know it? That's a thing that has to be that I thought should communicate it, because you essentially lose the purpose and the syntax in grammar. I mean Sanskritam, if you don't understand what exactly was mentioned. So we try to analyze this. For example, Shita and Hima. The word Shita could be translated again using different syntaxes. Sashay or Abhedya is what is a sutra mentioned in the Kashakursana grammar. In the Paniniya grammar. The Paniniya grammar says Sa and Sha can be interchanged. So Shita becomes Sita. Sita means a line, a demarcating line. And La means La Tatra Rachayana Charyaha. La means a chemical reaction. So Shita is definitely cold, because Shita Rutu we say it's a cold, is a winter season. But then the cold of the biological environment, coldness in the biological environment, is a result of a chemical reaction which is rate limiting exile. See, that's how, if you understand, you figure out that, okay, Vata is a biological environment in which predominantly endothermic reactions are taking place. And that's why, because all rate limiting reactions are endothermic reactions in the body. So these endothermic reactions, they're taking up heat of the body, in the surroundings, and then they make the system cold. And then the system gets cold, apply again laws of thermodynamics, the 0th law, the first law of thermodynamics. You essentially, water binds to the system, and hence the system becomes dry. It's not that the system is dry, but the system becomes dry because the water is unavailable. So that's how, friends, the environments, as mentioned in Vata, they correlate to certain organoleptic and buffer properties of the biological environment present in the body, and they classified them into Vata, Pitta, and Kapha. They gave even the characteristics. So when I started understanding the fundamentals of Ayurveda in this manner, we started digging deeper, really. I'll just translate quickly this one, the word Hima as well. Hima means ice, right? And ice makes the system cold. But Hima, again, you apply the Kashakursana grammar principles, so Paniniya grammar, it means Haya ho Rabedya. Ha and Ya can be interchanged. So Hima, interchange it with the H in Hima or Ha in Hima, interchange it with the Ya in the, and then interchange with Ya, you end up with Yima. Yima means coming close, condensation. So Kapha is an environment also cold, but biological environment becomes cold by condensation reactions and not but because of endothermic reactions. So that's how I slowly started understanding the depth of this Ayurvedic references, friends. It's going to be a huge long topic which we are going to discuss, of course not now. It's a part of my 545 hours of lecture for MBBS and or PharmD and or the BAMS doctor, so that they become better MD physicians with a PhD background. So that's a MD PhD program is being rolled out very soon, and we're going to come up with that. Help people understand the original essence of translational Ayurvedic theoretical knowledge, and then later do experiments and either confirm or refute it. But then the key is that not just the Ayurveda mentions about these 3 concepts. As I mentioned earlier, it also mentions that Ashitish Chatulakshara Samritam Swastha Ityabhidiyate. What it means is the body has about 8.4 million biomolecules in the body. Ashitish Chetwa is 84. Laksha is lakh. So 84 lakh chemicals are present in the body. And I was really baffled when I looked at this data coming from these books called Rasendra Sarasanga, Rasaratna Samuche, and several other books on alchemy and chemistry, friends. They already classified all of these 8.4 million biomolecules present in the body into water soluble. They say Jalaja Navalakshani, based on their solubility profile, they're saying 900,000 chemicals are Navalakshani, or 9 lakh chemicals are Jalaja, water soluble. Sthavara, Vimshati Lakshani, non-soluble or stable compounds are 25 lakh chemicals, and fat soluble, Ghrita, Chaturasya Lakshani, and so on and so forth. There's an entire solubility profile, given in about 16 verses, friends, of the biomolecules present in their body. And frankly, biochemistry is, I love biochemistry. 7 million compounds are detected in the body as of now, of which we know the solubility profile of only 9 lakh chemicals as of now. We still have to do a lot of data mining. We need to still classify, characterize these chemicals, understand what they are doing, and so on and so forth. But I was amazed looking at this data. Of course, it could be theoretical only. Is there a practical way of understanding these concepts mentioned? Yes, there is. We need to create models. That's what we've been trying to do. And just summing up, 8.4 million chemicals that end up with Dvam Trin Sahasra Kotichas Rasayanani Karshani, which means 32 trillion chemical interactions are happening because of these 8.4 million chemicals. And they are all classified into 21 broad categories based on their 7 of these categories from these 21 broad categories guide about 11 trillion chemical interactions, not just the chemical reactions, but also interactions. They guide the molecular pathways. 7 of them guide the metabolic, and the rest of the 7 guide the assimilative pathways, and they are called as Vata, Pitta, Kapha, friends. That's Ayurvedic biochemistry for you. That's very, very advanced. Very, very interesting. If you start understanding why the Vata, Pitta, Kaphas are given, according to me, these are the buffer properties of different groups of chemicals that act in tandem, friends. A bit of quantum entanglement is already taken into consideration with supra-quantum concepts of Ayurveda. That's what I would sum up for you now. Let's go to actual experiments that we did. What a typical Ayurvedic physician has to be doing is, because all of these 3 agents called the Vata, Pitta, Kapha, and they're not just 3, friends. That again, each of these agents are classified into 5 different categories. So there are 5 Vatas, 5 Pittas, and 5 Kaphas. The Prana, Apana, Vyana, Udana, Samana Vatas, Sadhaka, Alachaka, Ranjaka, Brajaka, Pachaka Pittas, Avalambaka, Kledaka, and so on and so forth, Kaphas, friends. So in all, 15 different biological environments are detected, and it is postulated or even documented in Ayurveda, these ancient scriptures, friends. These are documented, fully explained in a very, very scientific manner. Because you'll be amazed, anyone I would recommend to read, if this is interesting for you, read the Sutra Sthanam, friends. That really illuminate your brain. When you become like, you feel the Einsteinic moment in your life when you read the Sutra Sthanam, friends. So what Ayurveda does is, because all the physiological, immunological, all the processes are already classified based on VPKs, you use a certain technique called the Nadi Pariksha by applying these 3 fingers, put them on the radial artery. There's a method for that. Apply differential pressures, by pressure actuation, you're at 7 different levels. You're essentially detecting you end up with 3 fingers, 7 levels. You end up with 3 into 7, 21 data points on the right hand, 21 data points on the left hand, and each of the data point, the pressure point, corresponds to organ and certain organ system. So there are 7 tissues. You end up with Rasa, Rakta, Mamsameda, Asthimajja, Shukra, Dhatu, VPK proportions of these different tissues. And once you get this VPK 42 fingerprint, friends, you apply time domains, frequency domains, mind domains, at 24 different characteristics, as mentioned of the nadi by the ancient scriptures in Ayurveda, and they help you characterize Vata, Pitta, Kapha, detect VPKs in different 42 organs. And once you know that, because in Ayurveda, everything, as I mentioned, the foods, everything that you see, everything that you sense, eat, has a VPK fingerprint. That's the beauty of this. It's already given. So for you, most of the data science is already worked up in the Ayurvedic, because the lines of separators between Vata, Pitta, and Kaphas are already given. Once you have these lines of separators, you have to just generate data and then correlate them with the modern science. That's what we try to do. And what you do once you do this, then your algorithm, as mentioned in Ayurveda, should pick up those foods that balance your Vata, Pitta, Kapha. It's not that there's going to be a standard food, say Mediterranean diet that's good for your heart. It won't do good. For Mediterranean diet will be bad for Kapha individuals. Your keto diet will be good only for Vata individuals and bad for Kapha individuals again. Again, your millet diets is going to be extremely bad for Pitta individuals. And why am I saying this? Because I know the syntax of Vata, Pitta, Kapha, of these different foods. And because I know the impact of these foods on these doshas, as I mentioned earlier, that the balance of these three agents is leading to health, imbalance will lead to disease. So anything in excess is a toxin, as explained in Ayurveda. So ati sarvatra varjayet. Khatam. So that's why, friends, what we did was, the crux here being VPK profiling. We tried to create models of Vata, Pitta, Kapha in experimental animals, Wistar strain rats. What we did was we fed Vata aggravating, Pitta aggravating, Kapha aggravating diets to 3 different groups of animals respectively, and we fed the balanced diet to 2 groups of control group of rats, and then for 1 whole month. Why 1 whole month? That's another big story. I don't want to talk about that story because it's going to take 1 more hour. We already are through the 45 minutes, because I wanted to finish by 12:30, but I think I would need 5 more minutes. The organizers sell up, because this is what I mean. If you get this message, then you'll be motivated, and I'll be happy, because together we can work. So what we did was we correlated that at the end of 1 month, as mentioned in the Ayurveda, because each dosha gets enhanced or pacified, it requires dvipakshe, 2 pakshas. 1 paksha is 1 fortnight. So 2 fortnights are required for a certain dosha to get enhanced in a certain tissue. So that's why we fed these rats for 2 whole pakshas, or 1 whole month. At the end of 1 month, we isolated the hearts based on Langendorf method. Friends, these experiments were done in Moscow State University, and we did several times. We at least repeated them at least 29 times, these experiments, so that we checked if we were getting the same data. So we isolated the hearts based on Langendorf method, and then injected a voltage active dye, -2,4 dianep is a dye that we injected, and based on which the action potentials were captured through video cameras. And also we used a glass electrode method, which is sodium titrated, potassium titrated, chloride titrated glass electrode method we used to essentially detect these and register their action potentials. So I don't have to teach you action potentials. We all know there's something called the resting membrane potential. When there's an external stimulus, the resting membrane, the sodium ions, there'll be a slow influx of sodium into the cell until it reaches a certain threshold of -65. Once -65 is reached, then there's a fast sodium channel that opens up, leading to quick influx of sodium ions into the cell. And that's why you see the spike here. Okay, so you see the spike here, because at +5 to +15, the chloride ions also start moving inside. At +20 to +35 in different tissues, potassium starts moving out of the cell. So that's how you end up with a certain action potential. And at the end of the action potential, you have this refractory period here, of course. And at the end of action potential, the body uses up ATP to reverse this ionic or to bring about ionic homeostasis. So that's pretty much what the body does. You know what, friends? What was amazing was we found that under the influence of Vata, the Pitta, and the Kapha, the resting membrane potential was getting stabilized at 3 different levels. What does it mean? The The threshold membrane was across the tissues was pretty much, at minus 65 millivolts. Resting membrane definitely varied. It was minus 72, minus 90, minus 108, approximately this average. In Vata, Pitta, and Kapha aggravated or enhanced diet-fed rats, which means Vata tissues would need only 7 millivolts of equivalent external stimulus for them to elicit an action potential. If you give 7 millivolts to a Pitta or a Kapha, they won't elicit an action potential. So Vata tissues are pretty much hyperreactive. Now the Kapha tissues are like, they need 43 because the difference between resting and threshold in Kapha is 43 millivolts, friends. So you need a 43 millivolt of equivalent external stimulus for the Kapha hearts to elicit an action potential. It's amazing, isn't it? So if you supply an external millivolt, hypothetically, theoretically, it's absurd, but I'm just trying to help you understand. I'm just giving an analogy. Don't dig deep into the analogy, friends. So if you supply 53 millivolts of external stimulus, the Vata would elicit theoretically 7 action potentials, Pitta 2 and Kapha 1. If you supply only 7, I already mentioned Vata only is the one, Vata system is the only one that elicits an action potential. Pitta and Kapha won't elicit an action potential. What does it mean? Number one, it means that we have validated what is mentioned as Chalatva. The property of Chalatva is attributed to Vata. Chalatva means mobile. Okay, it's translated only as mobile, but applying different principles of Sanskrit sutrams, we could translate it as hyper-excitable as well. Why is a Vata tissue hyper-excitable? Because of this. And no one validated, friends. We were the only ones who validated this. This gives me immense pleasure because, for the first time it was done, and how beautifully our rishis, ancient scientists of India, friends, have beautifully classified Chalatva. They use the word Chalatva. They could have used, they could have used some other word, but they use the Chalatva because Chalatva word means, applying different grammar principles, hyperexcitability is attributed to the word. Hyperexcitable, why? Because the resting membrane is very close to the threshold membrane. So they require only very less stimulus and hence they are Chalatva. The Kapha is attributed by Sthira, very stable. Why are they stable? Because of wide gap between the resting and threshold, and hence the external stimulus should be more than this gap to elicit an action potential. So they're stable systems, friends. So amazingly, this is for the first time corroborated. And what we did was we further, did a lot of studies on this, created models of Vata, Pitta, Kapha further, checked their action under the influence of acetylcholine and adrenaline, and we sympathetic and parasympathetic mediators. And we found amazing things, based on these experiments. So to sum up, okay, the Pitta systems were characterized by non-reactivity, literally non-reactivity to either acetylcholine or adrenaline, friends. Amazing, isn't it? They don't respond to acetylcholine and adrenaline, which means you talk to Pitta aggravated individuals, you talk to them, reason it out, they won't listen to you. वो सुनेंगे ही नहीं आपकी बात, क्योंकि उनके मतलब बेस्ड ऑन दिस एक्सपेरिमेंट्स हम ये बता सकते हैं कि उनका दिमाग पे कोई एसिटिलकोलिन का एड्रेनलिन का असर पड़ता नहीं. But how are they eliciting action potentials is the biggest question, isn't it, friends? What we found was, we found 5 times more alpha-ketogluteric acid in the cytosol of the Pitta cells. 5 times more than Kapha and at least 3 times more than the Vata systems, which means the alpha-ketogluteric acid, we all know, is a part of the Krebs cycle. Right? It's so it is 5 times more. Iska matlab hai, it must have been so much, it must have been produced in such high quantities inside the mitochondria. It has escaped the mitochondria and come into the cytoplasm, which means that the mitochondrial activity is 5 times higher in Pitta than other systems. And alpha-ketogluteric acid, why is it so important? Because this has shown in several experiments, alpha-ketogluteric acid is another mediator that elicits action potentials, friends. So that's why Pitta systems elicit normal action potential, literally comparable to control group of rats, which are fed on balanced diet, but they don't respond to acetylcholine and adrenaline. उनको खाना खिलाओ तब वो ठीक हो जाएंगे, मतलब sweet खिलाओ उनको. मतलब what is happening is, friends, कि if you again translate into the ATP consumed, the glucose that is demanded, ATP consumed in Vata is 5 times more than Kapha. It's 2 times more in Pitta. So which means, because at the end of action potential, as I mentioned, the ionic homeostasis should be restored, which means sodium should be effluxed, chloride also should be sent out, Potassium should be brought back in. So that requires an ATP. So the ATP is used faster in Vata because they are hyperexcitable. Pitta is moderately excitable. Kapha is hypoexcitable, right? So the ATP accordingly is used in these three different environments. And glucose demand is highest in Pitta because Pitta is burning up glucose faster. All right. Vata is only hyperexcitable. It's not hypermetabolic, right? So the glucose demand is definitely high. Why? Because it is using up ATP faster. But if you look at isolated Vata systems, their hypochondrial activity is just as normal. Could be as normal. We have to do those experiments. But what I can say for sure is this glucose demand is only twice as much as Kapha. Kapha, because of its hypoexcitability, is consuming ATP less and demanding also less glucose. And hence, when you push glucose into a Vata environment, what happens is acylation reactions take place. When you push into the Pitta, so when you give more of Vata, glucose into the Vata environment, it produces acetaldehyde or acetyl alcohol, ethyl alcohol, and it gets acylated, friends. In Pitta environment, the glucose burns up faster, produces CO2, H2, and ATP. They're very highly energetic people, but they produce a lot of CO2 and H2O. When the partial pressures of CO2 and H2O inside the cells are high, their environment, the carbonic anhydrase gets activated, and from CO2 and H2O, carbonic acid is formed. Interestingly, amla or acid is one of the properties of Pitta environment. So it's only in Pitta environment that you have acidic environment, friends, in the tissues. So your Kangen water will work here, not for Vata or Kapha, because Vata and Kapha both are already alkaline, highly alkaline. Slightly more alkaline than normal. So, and also the way glucose is metabolized in Vata, Pitta, Kapha are different. And hence, if you push glucose into an environment of Kapha, glucose, most of it goes into storage, triglycerides, VLDLs, and of course lipoproteins are required. The body produces, but over and above, the glucose will go into the storage molecule. So it's like you take a piece of iron, put it in an electric environment. The iron piece gets electrified. But if you put the same iron piece in a magnetic environment, it gets magnetized. See, the same piece. So similarly, the Vata, the Pitta, and the Kapha are different. What are they called as? Buffer properties, buffer solutions, under the influence of which different molecules behave differently in different individuals. So that's the crux of your difference in pharmacodynamics and pharmacokinetics, friends. That's what I assume. So that's why the Vata diet is different, Pitta and Kapha diets are different. So your VPK fingerprint in different organs and different tissues is actually guiding the way the glucose is metabolized, the way is glucose stored, the way Pitta aggravated individual. You see why? Why? Because if you look at the data coming in nowadays, I pulled up several papers published on yoga. 27% of Vata aggravated at that particular point of time individual, it won't do anything. It won't do any benefit for him. You have to do the Vilom. You have to do the reverse differential nostril breathing. You need to breathe from the left and breathe out through the right. These are these are very advanced concept, friends. So that's why your yoga or fitness regimen or breathing exercises or pranayama, everything has to be properly personalized. And the key for personalization is the VPK fingerprint, friends. So that's pretty much what I wanted to say, because once you have this concept of polyscientific Ayurvedic advanced medicine in place and detect your VPK42 fingerprint, then will you be able to give appropriate diets, appropriate exercises, appropriate breathing exercise on these end stage, say IDDM patients, for example. End stage patients, we stick to the guidelines, and those who are excluded from the guidelines are the ones that we recruit in the stage 4 refractory diabetes, refractory hypertension, refractory kidney disease, and these are the patients who come to. This is one of the IDDM patient, friends, 4-year-old male with anti-GAD and anti-insulin antibodies, more than 2,000 and 48 and 2,000 respectively, after 2 years of our treatment based on VPK fingerprinting and giving them appropriate diet. Herbs also should be processed differently. There are 16 different processing methods given in Ayurveda, like you can give Arjuna or any herb in the form of a guti or a vati or a kashaya, arka, kshara, asava, arishta, and so on and so forth. There are different processings because vata environment, because it's highly reactive, you need to give something stabler. You cannot give an asava in a vata individual. You should give a kashaya in a vata individual. I mean, kashaya is a water extraction. Why? That syntax is already given in Ayurveda. I have not invented. I'm only trying to validate. So when I did this, when I give appropriate herbs, because each herb has its VPK fingerprint and its syntax, it's a specific organ that it acts on. And there is a certain method in which each of these tissues get balanced. And what happens when you do that, it takes little time, but then after 2 years, we were able to demonstrate how anti-insulin antibodies came down, the GAD antibodies came down. Not only this, his dependence, he was on 60 units of insulin at the time of 4 years, and by the time he became 6-year-old, we reduced it to six units. Not only just this is a heart patient, friends, vata mediated heart disease is vata shuddroga. This is the block gone after 6 months. There's a pitta mediated individual. The treatment methodology, the food, the exercise, everything was different. This lady, 54-year-old with concomitant diabetes, was actually on our treatment because she already underwent a sequential graft 18 months before she came to my clinic way back in 2003. She was given only 6 months to live because her EF fell down to 27%. And there were severe blocks again. LAD was blocked completely. After 3 months, it slowly opened up. 6 months, completely opened up. She is still living at the age of 73 plus. I mean, that's what we were able to do in end stage patients whose EF is so low. If EF is less than 30, friends, there's nothing pretty much the modern science can offer. Those are the patients who we recruit end stage heart diseases. I'm showing these are multiple blocks gone. There's a kapha individual. So kapha based remedies were given. So the diet was different, the exercises were different, the formulations were different. The way we processed were different. This is psoriasis, 20 years. For 7 years, his kidneys got calmed off. He was on dialysis alternate day, 3 times in a week for 7 whole years. 9 months after a treatment, completely gone. 18 months after a treatment, his dialysis, we weaned off his dialysis. Okay, we work with the doctors, friends, and then end stage cancers, B-cell lymphomas, brain dead patient, completely normal now, with brain regenerating. These are the regeneration technologies that we deployed in those patients who are rejected by the modern medicine. So this is like a vitiligo before, during, and after, friends. This is what we achieved in more than hundreds of patients, friends. This is, you name the disease, we cured it by God's grace. End stage again. There's another patient, ulcerative colitis, Crohn's disease. Crohn's disease after 1.5 years, completely stopped all for MMFs and all drugs that he was also on MAP therapies. Everything stopped after 18 months, and we slowly were able to demonstrate supreme efficacy in different diseases, friends. 28,000 plus patients, allergies, asthma, type 1 diabetes, autoimmune disorders, autism, various chronic diseases. We did a lot of work. And why am I saying this is, friends? All this is now captured. This is a very, very advanced algorithm, I would say. And there are several people claiming to be detecting Vata Pitta Kapha. I'm not against any one of them, but I'm for anyone and everyone who has data. And because we were able to model Vata Pitta Kapha in experimental animals, we were able to correlate precisely Vata Pitta Kapha and then correlate that to the heart rate variability algorithms. And we were able to come up with these advanced algorithms that would detect your VPK42 fingerprint, friends. And I have included in a model because of my love for biochemistry. I also deployed a certain mathematical model that, with the pulse photoplethysmography method, uses certain mathematical algorithms. I just quickly gave a rough outline of how I have arrived at these how I have arrived at these algorithms. We use several, I came up with at least 17 different machine learning algorithms and deployed them into this model. And this model gives a rough estimate of your blood glucose, HbA1c, and lipid profile, friends. First of its kind in the world. We're still, getting it validated. We're at the last stage of getting this clinical trial approved. And Mujib, my dear friend, is really helping us through get all this documentation done, because we wanted someone neutral to be unbiased and then, guide us, and also, grill us if necessary and when necessary, because scientific fervor will start with your death. I mean, which means you should be grilled so much by scientists that you're covered. I mean, you cover all aspects of scientific technology, friends. This is what we did, and there's a lot again. That's again another big discussion. So the idea is that when we were able to detect these VPK fingerprints, and based on which customize each and every solution that you give. Even pharmaceutical drugs can be given. Say, for example, if someone has pitta aggravation, you give him metformin. That's going to be good for him. If diabetes is pitta mediated, metformin is the choice of drug. If if a kapha mediated individual ends up with diabetes, definitely you need to use dapagliflozins and other gliflozins, friends. Sodium, salt receptor blockers, the glut receptor blockers in vata individuals, and so on and so forth. Again, because what we are doing now, if you look at this point, friends, again coming back to data science, machine learning algorithms, you could arrive at this point from this point in 3 different pathways: a linear algorithm, a non-linear algorithm. All right. So we are now discussing in modern medicine, which is definitely, I have immense respect for that. We are only discussing these values, friends. But how did a certain individual arrive at this value, whether it is vata mediated or pitta mediated or kapha mediated, based on these 3 algorithms or mathematical formulae, is something that we need to know, because we know, you all know too, that everything in the body, all biochemical reactions in the body are reversible. Only if you knew which path the body took to arrive at a certain point from a certain point. All right. So if you know the pathway, you can always reverse it. That's the key here, friends. That's all I can say for now. And that's what, when we were able to, first of all, we modeled vata pitta kapha. So we have a clear-cut data points of what is right data, because right now what is happening in Ayurvedic research is completely wrong, and also fake data is being used to mine, and that's very, very dangerous. That's definitely dangerous. Why? Not only for the human population, but also for India as a country. If you apply fake data for mining Ayurvedic parameters, you will end up with wrong algorithms, and people will say, of course, the Westerners are not foolish people, friends. They will figure it out for sure. If you're not fast enough, and if you don't work together, that's what I could say for sure. India was never invaded by foreigners. We have invaded ours because we're so intelligent. We cannot allow anyone to invade us, friends. We have done it ourselves. That's what I would say. And by the way, what happened? Invaders were also so they become so mesmerized that they got integrated, friends. That is Bharat, friends. That is what I would say is also applicable to data mining. If we properly are able to understand extract hidden insights of parameters, and translate that into knowledge discovery in Ayurveda, I think you're going to improve your clinical decisions. Because what happens is that in a kapha environment, going back to the old slide, if I have it here, 1 second. Oh no, sorry, we lost. Yeah. So if we are essentially 1 second, please. So if you remember the first slide that we talked about, in which I showed how endocrinologists have a different parameter. So now I clearly can understand why they have different parameters, because they investigate different individuals. A kapha individual, because he was hypometabolic, needed only less glucose. That's why American Diabetic Association is right. All right, they're also right. Why? Because they possibly have investigated kapha dominant individuals. The Veterans Affairs must have investigated who are pitta and slightly vata aggravated patients. So they're burning up glucose faster, they're using up glucose faster, so they need more glucose in the blood. That's why they could not resist. I mean, they not only tolerated, but they really enjoyed HbA1c levels of for 8.5 to 9.5. All right. So, and your kapha, pitta individuals, vata individuals, they're only hyperreactive and were able to process glucose faster. They were pretty much, essentially trying to One second, I'm just trying to put this slide again, friends. Then slide. Okay, I lost this again. Anyway, friends, so that's the point I'm trying to make. So this data, if you don't have the right data, friends, you'll make, because in data science, there's something called as, data out. I mean, the output will be junked, and then, your algorithm will be wrong. So that's a hazard of this. So what I would sum up is, there is immense benefit that our understanding of Ayurveda could do to modern medicines. Just like I said, how diabetic drugs could be even classified based on, we're trying pharmacogenomic methods to personalize their drugs, drug prescription. I would strongly say that if we had approached this pharmacometabolic typing based on PSA or based on VPK42 fingerprinting, we could arrive at better drug prescription to different individuals, and based on which you can use the right drug for right individual and then have less side effects, friends. So this is what I wanted to share today with you, and I really thank the organizers for bearing with for my delay in joining you people, and thank you very much everyone for giving me this opportunity to share.
Thank you, sir. Thank you so much. So I convey my regards to Dr. Ravi Shankar, and also requests to Dr. G.D. Gupta, sir, to please conclude this session. Sir, please unmute yourself.
Thank you very much, Dr. Mehanji. First of all, I am thankful Dr. Ravi Shankarji to provide the opportunity to our faculty members, participants, and overall throughout the pharmacy profession. So during the talk, you have considered very, very important parameter, including the latest technology of the AI and MI, linear as well as the non-linear of the algorithm. And we all know that Bath, Pitta, and Kapha, these play very important role in our life. Even that any types of the allergic condition, and sometimes we have some of the severe types of the disease, except of the fractures. I hope heal that each and everything is the basically because our body is the completely balanced with the system of the Bath and Pitta and Kapha. So you have focused and highlighted each and everything very, very in a scientific manner, and you have also included that how we can our modern medicine system and their limitation you have also given, current problem in the modern medicine you have also included, as well as you have included each and every parameter which are required for the health profession. So thank you very much, sir. And I hope that this types of the talk it will be the useful to each and everybody. And what are you performing in the, you have also given one of the slide is the yoga, meditation, like each and everything. So each and all types of the things have their own value. Anything in the excess and without maintaining the, you can say, property or value or something is the wrong, bath pit obviously it can be increased. And when you have taken the card in the morning, evening, and afternoon and the night, so all four conditions it will be the change of the value of the your throat problem maybe take place. Sometimes some of the other things maybe take place due to this reason. So take care and thank you very much, sir, for accepting our invitation. And you have given the lot of information in a very, very technical manner. Thank you very much for our whole team. And I hope that in the coming days, such types of the talk we will organize in the regular mode. So thankful to the whole of the team of the IQAC and our FDP program organizer to provide such types of the very, very eminent speakers, those who have their own value in their particular subject. Thank you very much.
Thank you very much. Thank you.
Thank you, sir, for such an
joining.
informative session.
So I request to Dr. Kamya, Dr. Shridhar is also joined in this session. So we can continue with Dr. Shridhar, sir.
Thank you, sir, for such an informative session.
Thank you, Dr. Ravi Shankar, sir. So I request
Thank you.
to
Thank
Dr.
you, Ji. Thank
Sridhar.
you very much. Thank you.
Yes, sir. Dr. Sridhar. Good afternoon, sir.
Sridhar sir, good afternoon.
Yeah, good afternoon. Good afternoon, sir.
Good afternoon, sir.
Good afternoon, sir, and welcome, sir. Welcome at ISF College of Pharmacy.
Yeah, thank you for the invitation. And it is very good.
Sir. Thank you for joining us.
So for the next online session, our speaker is Dr. Sridhar Yashamena, Artificial Intelligence Scientist, Boston University, USA. Dr. Sridhar is a highly accomplished scientist and leader with over 24 years of expertise in global clinical research and development and pharmacovigilance across diverse therapeutic areas in pharmaceutical and biopharmaceutical industries. His career includes leadership roles in renowned organizations such as Novartis, Hitro, Vocard, Bharat Serums, Vivo Bio, Cytec, and GVK, making significant contributions to drug development and drug safety. He currently serves as an artificial intelligence scientist at Boston University, USA, where he combines his passion for innovation with his deep knowledge of clinical pharmacology. Dr. Sridhar's unparalleled contributions to the pharmaceutical industry, combined with his forward-thinking approach to artificial intelligence and healthcare, continues to make him a driving force in the advancement of clinical research and pharmacovigilance worldwide. His topic for the session is Artificial Intelligence Applications in Clinical Research and Pharmacovigilance. Now, I would like to invite Dr. Sridhar Yashamena for today's FDP lecture. Sir, over to you.
Yeah, thank you, Dr. Kamya.
Thank you, sir.
So I can. Are my slides visible?
Sir, yes, sir.
So how can I make into slideshow?
Sir, press F5.
And the entire faculty, and it's very good to interact with the pharma faculty with this new concept of artificial intelligence. So as you all know that India has been distinguished as a pharmacy of the world, and you are the main pillars behind generating the pharma professionals across the globe. So a big namaskaram to all of you, the faculty members of pharmacy. And this India being now the third major pharmaceutical production by volume, and is expected to reach around 80 millions, 80 billions from by the end of 2025. So in this presentation, because all are subject matter experts, I would like to take more advanced level of information for the various means how the market survey reports and what is the forecast, and how this artificial intelligence is helping or helping the drug development and the drug discovery process, also in the drug safety, which are the regulatory driven approaches, mainly in the from the scientific focus, as well as the adverse effects for the drugs, and the how it is a boon for the patient safety, which is more miraculous. Mainly the current timelines handling numerous amount of patient data from the conventional point of view is very challenging, because it's now became a big data approach, where AI like technologies, means these novel technologies, as potentially has changed the landscape of complete drug discovery from the development and also from the drug safety post-marketing point of view. So saying these brief words, I'll go into the, because it's a lunch time, and I'll give you the correct message, what is happening in the industry and what is being developed and where are the critical challenges and how we have to overcome. So primarily, just a disclaimer, that is all my personal opinions and not related to any of my university or from the any of the my previous organization which I have worked with. Yes, definitely. Some of the images and the slides have been developed with artificial intelligence tools and with permit. So firstly, I would say that the global marketplace for the major companies which adopted artificial intelligence in the scope of drug discovery and development is tremendously increasing. So as you all already know, the CAGR, the compound annual growth rate, is 31.6%, which is higher than the other, mainly the return on investment, ROI, where we calculate by the economic terms CAGR, which is highest in this area. Of course, in the clinical trials and in pharmacovigilance also, the CAGR is around 25 to 35%, and whereas petroleum industry is around 14%. So you can understand the importance of the compound annual growth in this area, as the products or the drug development market is expected to reach around $400 billion by 2027, which is just 2 years. Of course, but the growth of this means mainly impacted by various other challenges like cyber security crimes and also some viruses, ransomware virus in the technology front, not from the human point of view, which acts, which were the hurdles during the development process, where most of the companies have invested very huge amount to get into a proper AI related output in Asia Pacific as well as the Western world. So saying this, even if you see the area of pharmacovigilance as a market potential, this is officially released market value report, which is going to be around $16 billion by 2030. So this also shares the amount of value, because every marketed product need to be abide to the regulatory regulations across the globe, and we have to maintain that until the life cycle of the product is maintained. Saying this, I'll go into the complexities. As most of you know, the conventional process of clinical trials from phase 1 to phase 4, there is a good type of innovations happened in phase 0 also, totally 5 phases of trials, where the intricacies and the challenges associated with the design of the study, the implementation and management of the clinical trials, where in a nutshell, I want to present with this slide, where various factors like the systems that we use, clinical systems, and the clinical supplies for the various reference and the test product, and also other supplies, and also the central labs, because to maintain a uniformity of the lab investigation during the clinical trials, we have to have the central lab facilities. So there are many NABL accredited, CAP accredited laboratories in order to ensure the values are acceptable by the regulators. And also there are very good trackers which are developed by the various other tools, means apart from artificial intelligence, which are being used. And also to the supporting to the pharma companies, there are many CROs which are handling the various expertise area of services, so that the multiple CROs will be involved in one clinical trial. And overall the communication channels and the certificate of analysis and the data management, big data that we were discussing, and the operational challenges mainly, means we categorize into 6 categories of challenges. One is operational challenges during the handling of the clinical trials, and also from the sourcing from the various resources and the project teams and the studies teams at the site level, and also the supply management of the proper condition, temperature control and everything, and the overall functional capacity. So in these various challenges, if you see, the trial complexity has moved from if you know, the paper-based challenges, which was more time-consuming, and where many other other manipulations, everything were possible in the paper-based. But however, the sponsors are the pharmaceutical companies and CROs came with clinical trial management systems, which are associated with very good audit trials, and also very controlled systems, from where the data transparency gaps were overcome. And also from the multiple CROs involvement in the multiple CTMS, we have an ability to track various things. Means the systems are such a way that the one clinical trial, global clinical trial, which is happening in India, is also been evaluated. The lab parameters, ECGs and X-rays have been evaluated by the cardiologist and the other people from the Western region, or US and the Europe region. So this way, an independent review of the patient data has been obtained, so that the accuracy and the authenticity of the trial data was more evolved. So now that we have got the agile systems of the cloud-based technological solutions, and the complete mindset of the clinical trial or the pharmacovigilance, or the entire drug development process is changed from post-COVID. So pre-COVID was the conventional method. Post-COVID has given a lot of learning lessons, where if you have any pandemic or any site of other diseases which evolve all of a sudden, where contagious related things, the development of a good vaccine or any product for the which are safer, so those type of things we have to overcome. So hence the usage of this powerful visualization techniques of artificial intelligence or insights or the analytics is helping to overcome understanding and managing the complexities. So the first major thing is the regulatory compliance. So gone are the days where the different regulations were existing, but ICH has brought all the harmonization across the globe, where 90% of the regulations are streamlined to process the requirement. Even the regulators are discussing among themselves before approving any drug for any clinical trial or for the marketing authorization. And same way, certain diseases where you can consider as the product may be considered in the class of first line therapy in US and second line in the Europe, the designing of the protocol becomes a more complex where in the clinical trial. So in this schematic strategy, where to understand both regulations and both line of treatments, it is very difficult. Hence, the regulators, after the discussion with the regulators, we have implemented certain artificial intelligence tools, so that the tracking of the information from the both first line therapy and the second line therapy is also possible in the same clinical trial. So that way we are minimizing the timelines associated with the clinical trial execution. So also the data collection from the various global trials, means almost more than 10,000, 20,000 patients data in a global studies, which need to be evaluated. So it is very difficult for a manual intervention with the individuals to, so there may be a lot of errors or the connected factors between the various teams that are operating. So these complexities were being reduced in introduction of these AI-based tools. So one such example I can mention you. So during the informed consent process, where in the global, so we are to obtain the information from the various sites across the globe, means 125 countries. We have to obtain the information of how many patients recruited at each country. So it is time consuming and the timeline difference, everything. But we have implemented a digital pen concept, which directly, as soon as the patient signs with the digital pen on the particular site, directly from the docket station through the intranet, it will interface and our dashboard will be updated with the wherever the patient has been enrolled. So any country, as soon as the patient is enrolled, we will have the recruitment status as soon as possible. So such simple, so it is useful. And this collaboration and communication even used for the site selection process, site feasibility process, patient recruitment process, until the last patient, last visit of the subject in the trial. So overall, apart from this, we can analyze the interim, means it's not that after 10,000 patient recruitment, we analyze the risk of what exactly the output of this. So most of the studies failed, or many of the previous 2 decades back incident, if you remember. So those will not be there when we have this type of interim analysis. So what we have put an algorithm in place where every 1,000 subjects, we have in a complete elucidated the data which is obtaining, what is the risk, what is the primary outcome or the primary endpoints and secondary endpoints, whether the trial is meeting or not. So that a gradual progress of the study, we will be also understanding that the products have been, the overall outcome of the study is going in a right direction. Otherwise, whatever the necessary risk mitigation categories that we have to implement, we will be doing that. So that way, this type of technological driven has saving the cost as well as bringing the more compliance output from the clinical trials. So this way, till the closeout of the. So these are the various factors which attribute for the complexity, as we discussed: the patient outcome, the patient enrollment, and the recruitment category, where also on the regulatory requirements, whether we are meeting it or not, and the design and the protocol standards, mainly the investigational product, the specifications of the investigational product and the proper usage, the compliance of the drug usage, IP products with the patient, everything will be tracked in a proper system-based clinical trial approach. So because this is multinational, multi-center and big data volume and all those safety issues, so non-serious cases, serious cases, any SUSARs also will be identified immediately. If you remember, this means based on the adverse event reporting category, once the pharmacovigilance guidance have been implemented even in India in 2018. So post that, the major adverse events were be easily reported across the globe. So gone are the days where the thalidomide issue or other antibiotic issues where large number of patients have been affected. But now even recent NDMA factor or any other thing, the immediately within single digit or 2-digit patients get exposed with this type of adverse events, immediately it has been noted and the global was warned about such incidences. So the patient exposure was minimized. So that is also one of the utilization of the technology in reporting. So same way, even very ethical standards maintaining or the evolving the landscape of entire the life cycle change of the clinical trials is maintained. Yeah, now this implementation of AI-driven technologies has brought efficiency, and optimistic every step involved in clinical trials as well as in pharmacovigilance, which is bringing the patient-centric approach to the whole process. So the patient-centric in the sense, the safety of the patients. From an instance of reporting of the patient-related clinical data or from the safety data, which made interactive through from a speech to the text conversations, or quantifying drug-related harm, which can be enhanced through real-world data. So I'll show you an examples how we have come in the next stage from various vulnerable diseases, or we use the devices linking such data with e-health records, which are maintained by many of the hospitals, and novel way of ADR management solutions, which participated by the patients for the quality of healthcare while AI propels it. So overall, if you say that artificial intelligence is essentially an what do you say, the ability of a computer software to learn, computer software to learn rather than merely carrying out an instructions. It has been we can say that pre-programmed, or the algorithm approach which the drug discovery itself has minimized the target candidate identification, and also where you can get a promising molecule there. We can see the next slide there, where we can bring the libraries of potential drugs. So gone are the days with the conventional drug development process. So most of the future drugs will be with the AI-based approaches for the new diseases or the existing complicated diseases. We will have better molecules in future with the more safer products for the humans. So this is a very simple example where artificial intelligence uses various platforms, and we will see that. So the mainly this AI is achieving all this currently the available languages or the tools that are there in the process are process automation and interactive communication or reasoning. So for which, as you know, the languages, C language or any other languages. So for AI, these are the tools that are being used to get the appropriate outcome or the purpose. Based on the purpose, we have to use that tool. So like in statistics, based on the data that we have, we go with the student t-test or ANOVA based on the number of groups that we have. So how we differentiate based on the data type, a statistics test, and based on the purpose of the output, how we do this other programming of the tech related. In AI, these are the various tools that are being used currently for the development of any automation. So mainly for robotic process automation, you can use RPA and intelligent process automation like IPA. This will be used for a process automation. So which is, it's not a simple decision to make, but if you have a repetitive predictable task, this type of tools to be used. Whereas if you are obtaining an outcome of some analysis of a text to the voice or generate some text to the voice, NLP and NLG will be useful, which will gather the knowledge and which will share the interactive communication mode. So already many of you are using other tools used by the in the Google free chatbots, everything. And if it is a reasoning, why it has to be, we have to recognize in a pattern, and we have to recognize and enable a self-learning approach. Then we have to use the machine learning and artificial intelligence. So not only this, there are other tools which will be used for the development in the like Python or any other SQL related thing based on the output that is required for the in the drug development or in a clinical trials or in the pharmacovigilance. So here are some of the opportunities where the AI application in the healthcare, that is the pharmaceutical industry, and developed in the recent years, mainly from the digitalizing, means from the overall part for the hospital, means like you have now mainly you can use the entire digital technology to monitor the home, same way the clinical trial can be monitored, and also the precision of one individual health records, the progress, how it is happening. So everything can be documented. And however, means now you have AI enabled in the mobile phones, mostly, or maybe you are already using it. So you can obtain your health record, or mainly the heart rate, blood pressure, and the basic things already been incorporated in many AI developed tools. And even in the industry, when we are minimizing the more tedious job activities, we are also using these AI related applications to minimize the literature search, or doing the reporting aspects, electronic reporting aspects of the trials. So in the drug discovery and development, which was conventionally a slow process, like all of you know, the high throughput screening and serendipity, everything in the target identification. And from there, the human-led designs and in vitro studies, in vivo studies, and then preclinical, then clinical. And this was a conventional 10 to 15 years of process in the development. So the many companies where now there is no means I would say that the rate at which the drug discovery has completely declined is because of this tedious long duration of the activity. And also we're not able to predict the safety of the product, mainly the toxicity and other aspects of the product, which really has impacted drug discovery labs to close down in many ways. But in silico modeling, or simulation models that has been developed across the industry, has fastened the overall process, which analyze large amount of data, first thing, and also gives an insights of the drug discovery, mainly by predicting the efficacy and toxicity of the potential drug candidates, which will be useful for in a long term. Because unless we the failure of the studies in the phase 3 study, until the efficacy was completely not statistically significant. So these simulation models, or these in silico models, have evolved so that the target identification and also a promising potential drug candidate with good efficacy and safety has been proven. So these are the various other tools that has been used, mainly as we were discussing. So the machine learning is one such algorithm which analyzes large and complex data sets, and to identify patterns and relationships that may not be apparent to humans. In drug discovery, the machine learning used to identify these potential drug candidates and predict their efficacy and toxicity and optimizing them. And there are other things like deep learning, NLP, which computer visions and computer visualization. The main important thing is visualizing the images and the identifying pattern is one such way where the computer visualization is helping us. And computer vision is used to analyze all these conventional method of screenings and also development process as we have overcome. So in that way, these are the already market available tools which are from the public portal. There are other things which are still in the development startups, whereas atom-wise, in silico medicine and benevolent and recursion are the majorly which are being used. And this algorithm gives a virtual screening and identify potential drug candidate. And this algorithm analyzes the 3D structure of the proteins, and they predict the different sequences and allowing them to make it more high accuracy of the protein, which elicit the required desired therapeutic effects. And also in silico medicine, which is other company which AI powered identification of potential candidates they have doing, and there are supported by scientific literature and clinical trial data, and this proven to be the optimizing the correct drug designing. Same way, whereas the benevolent AI is also in such way as identifying potential candidates and in screening of this molecule. So apart from this, there are many other indigenously in India, they are developing these type of screening molecules, selecting various therapeutic areas, and it is still under the process. So they have not come out further. So one such example in recent years, which has developed by the in silico medicine and development a new candidate that is idiopathic for pulmonary fibrosis in the lung diseases. And this crema was designed using in silico AI powered based development and shown to be effective in clinical preclinical studies. And this is the first example of such candidates which happened in 2020. And the clinical development is in process. So because it's a first molecule and extensive process, and the guidelines are also not available at this moment. These are the one other challenge for the AI developed products. So we have to means the regulators have been developing these guidelines for the products which have been developed. Same way, Autumn was also empowered to get these potential candidates for the rare diseases like and also cancer and Ebola type of things. And they have currently the product that they have developed for this disease, this therapeutic area. They are very effective in the preclinical studies, but now in the clinical study phase. So maybe in a couple of years, we come with a good output of the data that is obtained for these drug candidates. Saying this, I will move into the pharmacovigilance part, where AI has a very good potential of because from last 3 decades, we have good safety information on the most of the generic products, right? So where we identify what types of the known, means the listed events, what we say are expected events, which is already known. Or there in the means because in the clinical trial, it is in a controlled condition where we were not able to get an unexpected adverse event, like SUSAR may not be obtained. But in a real world situation during the clinical practice, because every patient has different habitat, different food habits and the other climatic conditions, and also drug-drug interaction, comorbid condition, concomitant medication that the patient takes. The safety of various other during the routine phase also been developed in these 30 years. So saying this, so this AI is helping to analyze the data which is already obtained, because now we have this data in ultravigilance and Vigiflow. From there, we are getting this information, and this streamlining this in the pharmacovigilance system to bring patient information closer to the healthcare services. So by this, we may not able to repeat or take the same class of drugs, or we can minimize which are the moieties in the core SAR, structure activity relationship of any chemical moiety. We can understand these are the pharmacophoric group and these are the toxicophoric group. So we can minimize the toxicophoric group and evolve with a safety information. So because the drug safety or the pharmacovigilance being is one of the top 5 leading cause of death as mentioned by the health authorities. So apart from the disease progression and without knowing the safety of the products, if the patients have been prescribed and these undesirable effects are being patients are going into the morbidities, which is very significant. Sorry. So AI in pharmacovigilance is also developing many automatic task management from the identification, case processing, as well as from the quality review point of view. So any case information that obtained through telephonically by email, the AI is helping us directly entering all this information into the various safety databases that we use in the industry. And these skills are being developed. So manual intervention is reduced so that all information go into the safety database. From there, this safety information of one particular product will be enlisted as a line listing. And from that, we prepare the development of PSURs, periodic safety update reports, PEDARs for the US FDA. So these type of reports will be automatically generated so that the human intervention is reduced and more quality and the more precise information on the safety will be elicited in this process. So if you see this AI has a potential to improve, as we discussed. So this apart from the pharmacovigilance data that is enabled here, more efficient and accurate analysis of large and complex data sources by detecting safety issues earlier, and also known in terms of signal detection, so and signal refinement and causality assessment, so whether it is related to the drug or not, or whether the patient demographics or comorbid conditions really affect. And more importantly, what I am seeing is from this, as we mentioned, is last 3 decades information. We need to have a predictive analytics, where the safety information of the existing class of drugs, existing products is well known. So from there we can get better molecule output for the human race. So in real time, this AI is also helping in the healthcare systems, health systems, from electronic health records, as well as the amount of infection occurring in the hospital acquired infection and drug resistance models, and due to the polypharmacy comorbid conditions, polypharmacy conditions, this type of adverse events where due to prescription, off-label or diagnostics or any hospital-based, everything can be noted down in the real time and already been used in that. So one such example, when the Pfizer in collaboration with IBM have started this advancing drug safety through the intelligent use of the drug. So ADSIUD, which is again a platform developed for the Watson health system for drug safety, and where the AI algorithm for the large data sets has been used and detected potential safety information. So this platform already been shown to be effective and detected previously unknown adverse events and drug interactions. So this is the predictive analysis which we are concentrating. And apart from that, certain drugs are developed using biochipron technology, apart from biosimilars and other biologics. So it's technology derived products, and the same will be with the AI. So for those molecules, this type of tools which gives the additional safety information before it exposed to the human race. So overall, the ultimate aim of drug development is, we have to get a safe, effective products to the human race for their well-being and with very less side effects. So these are the few companies which are already been utilizing or implemented or invested a lot into the AI-based approaches. So whereas means everyone has into different therapeutic area, like in Pfizer into the non-alcoholic hepatitis and sclerosis way, whereas in Novartis working majorly on multiple sclerosis, because this is the highly prevalent disease in the Western region, and Alzheimer's, and also the carcinomas. And whereas our Sanofi is also into this process, GSK, and apart from this, also Indian origin companies which are not to this extent, but they are into the implementation of the AI-based approaches. So you can see, I'm sorry, maybe it is not visible, but there are many other companies which have been already been implemented, and we will be able to do more in the faster phase in the recent in coming recent years soon. So this opportunity of AI also enhancing the personalized medicine concept for the individuals, a tailor-made concept which we are discussing from when I started my graduation 25 years back. But still, now this will help us to give the personalized medicine approach, precision, but main importantly, the conventional clinical trials, which has many limitations in population selected, sample set that has been selected, and whereas in the same molecule in the real time is eliciting different. So this real time evidences, the real world studies, also will help through this AI analysis for the large case big data analytics, and to derive the drug safety and effectiveness. And apart from that, also there are existing drugs which are being off-labelly used for various other indications, that is drug repurposing. So this type of identification, mainly using these AI tools, will also enhance the more efficacious product more safer products to the human race. So some of these challenges which every time when we have a new technology, if you remember 2K issue, when the new computers have been evolved, where most of us are aware that the big breaking news that the all will be shut down because of the 2K issue. But however, the challenges will come, and there are good subject matter experts, and the teachers like you have given a good information on the basic foundation knowledge. So based on that, these are the challenges which occur in the technological related things also will overcome step by step because of the utilization of various newer techniques. And also I would like to say that there are various steps involved in this process of cracking the technology, understanding, and also potentially mitigating the each problem from the regulatory point of view, from technical point of view. Again, it should be an audit trail and it should be validated. Those are the important things which we have to more precise or more focused on when developing such AI-based tools. And many organizations are embracing with AI by mitigating many challenges that till now we were not able to mitigate. Now it has come to some solution. Many of the solutions have been there in the risk management. Saying this, so the entire drug development process, where the digital transformation of this drug safety information also in the process, and where the entire efforts that needed for the direct or energetic approach for mitigating risk and ensuring patient safety is clearly being fostered by various levels. Only the thing is, it's a matter of a couple of years factor where we will come out as an big means many universities have been started, and also I want to emphasize that this type of subject need to be also embedded into the curriculum in the all the universities. Like when clinical research and pharmacovigilance came up, all the curriculums have been changed by the various universities. This has to be implemented earlier the better, so that the understanding of this for the pharma professionals will also give innovative ideas in the own areas where we are working currently, or the new diseases that are coming up. So this way, the demand for the market overall will be means mitigated with this AI can be so AI has helped to identify potential issues with the drugs by analyzing large data sets of the adverse events and the healthcare medical records, and which also the next gen or CD models leveraging the predictive analytics, global real-time information, and intelligent detection and management of assisting human decision and improving safety and overall maintaining the benefit profile to minimize risk during the entire product life cycle. So the dawn in the AI in this will bridge the evolving demands of the market that would bring pragmatic approach through opening the doors for the opportunities in terms of infrastructure, AI-related labs, and the landscapes, and that also not only with the adverse event, that complexities which are there in the clinical trials, and the futuristic model which is there completely with the no more the traditional ones, would be completely efficient, predictive, manageable workloads by strategic merging of these tools in the real time. So overall, this will bring up a better products for the human race for the safety of the patients. Thank you everyone, and thank you everyone for giving this opportunity. I will be available for any questions or any discussions that we may have.
So I request to Dr. Kamya, please proceed the session.
Thank you, sir, for such a highly informative lecture. Now, I would like to invite our worthy director, Dr. G.D. Gupta, sir, to conclude the session.
Madam ji, first you can take queries, any question or any queries from audience side, faculty members or those who are connected in the online YouTube, please. So,
Sir.
no any question? Okay. So thank you very
Yes.
much. Thank you, Dr. Sridhar ji. And Dr. Sridhar is the delivered excellent lecture on artificial application in clinical research and pharmacovigilance. So obviously, in those days, this is the one of the big field in the pharmaceutical sciences also. Initially, he is the started and given the information related to the Indian pharma industry and worldwide. Indian pharma industry is the well known, and we are the occupy the third position by the way of the volume. So obviously, in the coming days, in the profession pharmacovigilance, it will be the grow very fast and have the lot of opportunity of the artificial intelligence. He has considered the market value of the pharmacovigilance, clinical trial complexity, ADR monitoring, understanding and managing of all types of the complexity. He also included the limitation of the AI in the pharmacovigilance as well in the health profession, and what is the application of the pharmacovigilance. He also considered in the ADR, because we know that pharmacovigilance student, they are the obviously doing the work on the database. So how we can represent our data and analyze accordingly. So each and everything he is the consider. He also given the number of the examples of the major pharmaceutical companies, those who are already included the artificial intelligence application in their production, in their data presentation, data maintenance, data collection, data analysis, and so many parts they have the included. So this is the good opportunity to the each and everyone. Either you are not, number of time I feel that if I am the person of the computer science or related to the like a machine learning, then I am the most suitable for the artificial. It is not essential that nowadays AICT is the providing the lot of opportunity, and recently one of the guideline given the AICT, any person they can be opt the any branch or one two branch at a time. You can do the BTech, MTech like that. And let us consider one more information is the given over here. One thing, any person is the BTech or MTech in the computer science, and if he has the capability to take some of the classes theory or practical in the mechanical science, then there is the no problem. So nowadays field is the open. It is not essential that you are the pharma people, so you need a one of the best to the best practice in the computer, then you can the only you can apply the AI. So I hope that in the today is the three lecture based on the artificial intelligence, related to the education system, related to the healthcare system, related to the pharmacovigilance like that. So today's whole of the all three sessions dedicatedly we have opted only for the artificial intelligence. So thank you very much, sir.
Right.
And I hope that this information it will be the helpful to our delegates, faculty members, and not only these who are connect with our YouTube channel, and those who will come to know about the artificial intelligence, they will take the benefit. And obviously your talk it will be the also motivate the faculty member ki any person can be the workout or involved in the artificial intelligence in their profession. So thank you very much, sir, and I am thankful to the our whole of the coordinating team to provide this opportunity to listen, just like your very dedicated and intelligent like of the speaker, and provide the opportunity to all the FDP members. Thank you very much. Once again, I welcome you and thankful to you for accepting our invitation and provide the opportunity to you can deliver the lecture on this platform. Thank you very much.
Sir, here I want to notify to all the persons that in United States of America, there is a midnight near around 3:15 p.m. AM. Sorry. And sir, thank you so much to Shridhar to sparing this time and providing this a very interactive session with all of us. We are highly thankful to you, sir.
Yeah, thank you very much. And namaste. Thank you, everyone who has provided this. And it has to be implemented in the academic.
Sir, there are two queries online. If you are able, then I can discuss with you, sir. So
Sure, sure.
one is what is the long-term impact on humanity with the utilization of this AI?
Long-term impact on
Humanity.
humanities. So we
Yes.
need mainly we are focusing on particular this thing in the better safer. So once we have, the AI related information will definitely lead to the more safer products. Now gone 20 years back where computer aided designing, which has obtained based on that the conventional process of drug discovery where it has overcome. So similarly, this will may means some technological related things, because if it is under regulatory conditions, if it is developed, like if you see UK also as a US has already implemented even the national security systems with the AI. So there is a controlled system so that any actions that is happening in the country, everything will be noted down with the security system. So that way, if it is regulated completely, the humanity can be maintained. Otherwise, that will be means every thought process, every perceptions will be different.
Thank you, sir. Thank you so much. So I request to Dr. Kamya to conclude the session so that we can take our next speaker, that is Dr. Gaurav Goyal.
Thank you, sir.
Yeah, thank you.
For the next online session, our speaker is Dr. Gaurav Goyal. Dr. Gaurav Goyal is currently working in the position of Deputy General Manager and heading Transdermal Formulation and Development Department at Zydus Life Sciences, formerly known as Zydus Cadila, Ahmedabad, Gujarat. He has total 15 years of industrial and academic research experience and worked with well-known industries such as Shilpa Medicare, Akums Drugs and Pharmaceuticals, and Rusan Pharma Limited. From academic front, he taught and guided students of ICT Mumbai, ISF College of Pharmacy Moga, and MET Institute of Pharmacy Mumbai. Dr. Goyal developed the first indigenously generic fentanyl transdermal patch commercialized in the year 2009, and later many more transdermal technologies were developed and commercialized. His focused research areas are development of oral thin films, transdermal therapeutic systems, implants, nasal sprays, lyophilized injectables, sustained release topical and oral solid formulations, etc. He is lifetime member of Indian Pharmaceutical Association, APTI, and Controlled Release Society, Indian chapter. His topic for the session is Quality by Design. Now, I would like to invite Dr. Gaurav Goyal for his today's FDP lecture. Sir.
Dr. Gaurav, good afternoon, sir.
Sir, your mic.
Yeah, good afternoon, everybody.
Good
afternoon, Dr. and welcome.
Yeah, namaskar, sir. Thank you very much. I am very much thankful to the ISF family. It's my honor to give lecture on this particular platform. ISF, I worked with ISF as a professor of pharmaceutics, and I am very thankful to Dr. Kamya Goyal for their kind introduction.
Thank you, sir.
Yeah. So I just present my slides.
Yes, sir.
Yeah. Is it visible? Are these slides visible?
Yes, sir. Slides are visible, but not in slideshow.
Not in slideshow. Okay.
Yes, sir.
Just a minute. Is it now in the slideshow?
No,
Hello?
sir. No, sir. Still, it's not in slideshow.
Okay. Okay. Thank you very much. Okay. Why the QBD is itself, everybody as a faculty, we all understand that it is a quality by design. So now it is quality by design itself. It means that it is a strategic approach to ensure the quality of the product throughout their life cycle. That we are ensuring by our approaches that the product will remain throughout its life cycle up to the shelf life. It involves integrating quality considerations into the design. Like we have to, during the development itself, we have to bring the quality into the product. We have to think about the quality, like it should remain up to this, remain stable up to the shelf life and during the different stages of manufacturing, packaging, and distribution. In QBD, we need to understand through the scientific knowledge that what are the processes involved in the manufacturing, and then we should understand the process, and then we design by understanding of the process we should design the formulation, and then we do the process control at the various stages of manufacturing. How we need to control the processes so that the product of the quality of the product should be built, should be increased, and these are the principles of QBD. But here today, I am going with the theme of the FDP. The theme selection is very good by the team. And today in this QBD, I am going to present that how we can do some QBD in teaching also. Like we can improve our teaching process by application, by our scientific knowledge, how we can improve. This one topic I am going to cover today. We can certainly improve the quality of our exam papers, and then the how to build, like whenever we are in practical exams, in practical classes, how we can apply the QBD in our experiments. And in nutshell, it will be QBD in pharmaceuticals. In the whole presentation, I am talking about the pharmaceuticals. This will be anybody from medical chemistry, pharmacology can apply all these things, can take the reference from these particular slides and can use these principles in their subjects. So today I am going to discuss more onto the pharmaceutics point with a perspective, but that can be applicable to all other subjects. So basically this particular presentation, the motive of this particular presentation is to sensitize our teaching faculty, because being a teacher, I was a professor of pharmaceutics in ISF College of Pharmacy. Before that, I was a lecturer in Institute of Chemical Technology, formerly known as UDCT. So now at various platforms, I taught also, so I can understand what are the difficulties of the teacher. But certainly we have to upgrade our teaching process so that we can able to give products to the industry. Products here means the students. We should train our manpower in such a way that it will be readily absorbed by the industries. Otherwise, it will be very difficult by the industries, that they reluctant to take the freshers. They want that the students should take some training from some other industries, small industries, and then they, after some 1 or 2 years of experience, they take the students on board. So that is very difficult. So if we have those capabilities in teaching, we have to upgrade ourselves regularly. The teachers, this is the very good platform where faculty development is there. We have to develop ourselves, and then we have to develop the students. So here today I'm going to with examples of this, and in the end of the this particular presentation, there will be 2 case studies where I want that the person who is giving the right answer at the fastest speed in a very less time, the 5 correct answers will be, those names will be like we can share on the screen, right? Here the QBD in teaching, everyone we are seeing here, there is a one object which is moving on the screen, right? So this is nothing but it is a chemical structure which is joined by the bonds, right? And this is carbon elements, atoms which are joined with each other, right? So if we see this particular chemical substance, we can see this, it is a same substance but having the multiple views. The view is different, like it is moving from every view we can see differently. So same thing, but because education is like the course of pharmacy is same, but we can give the view to this particular subject in different way to the students. How they need to think about that. So today's presentation is basically focused on this particular aspect. It is not necessary that we need to change the course curriculum of pharmacy, B.Pharm curriculum, M.Pharm curriculum. The same thing we can present to the student in a different way, different view, so that he will be able to get insight of the industry. We have to, because the main motive that we want that our students should go to the industry. Basically, we are preparing them from the industry for the pharmaceutical industry. Now, it doesn't mean like they should not, like in pharma industry, people are working and they should know the basics. So the basic terminology should be inculcated into them so that they will use those languages in daily life. So here, one thing which we are also seeing that all the dots are joined. So this join of dot, why this particular is this? You have, why we teach certain things through like definition of coated tablets, uncoated tablet to the student. And then in analysis, they are learning something else. In pharmacology, it is learning something else for the tablet. But certainly we have to join these 3 education, and then we have to make the question papers accordingly so that their complete knowledge can be judged. Instead of asking a definition, simple definition that what is uncoated tablets. Instead of that, how to design the questions, this we are going to learn in this particular presentation. So now the first example which I am basically focused in the complete presentation is the tablets. Like whenever we design something, whenever we cook something, like if you are preparing something in kitchen, then what is your expected outcome? It should be tasty. It should be some, if you are making some soup, you want that it should be some sour taste should be there, right? It should have a particular viscosity should be there so that it should not be very thin and it should not be very thick. So these all is like outcome, what we think for the soup itself in a kitchen. So in a similar way, tablets, what is the expected outcome in the tablet? That it should have the proper hardness. Why hardness is required? So that it should, when during the transportation cycle, when it is moving from, like the tablets are prepared, then the first point is like it has to be packed. In the packaging machine, it should not break. The corner should remain intact. And then during the transportation, it should remain intact and reach to the user, that is the end user, like to the patient, in a uncracked manner. It should not break in between. Correct? Similar is the uniformity. Each tablet should contain equal amount of drug, API. It should not be like this. One tablet is containing 2 mg of the drug, another containing 2.5 mg of the drug. So it should be uniform. So that is a major expectation from the whenever we are making some production, taking some batch in industry, it is a uniformity is at most required. Then it should be the bioavailability. It should have the bioavailability, right? In Indian scenario, we can say, like if it is an old product, we can show the bioavailability through dissolution methods, right? But it is like in vitro, in vivo correlation. If in vitro it is okay, then we can say it is in vivo, it will be good, right? And then we come to the stability. It should be stable up to the shelf life. And then it should be properly elegant. Definitely elegance is very important point, where the users or consumers will not take anything if it is not elegant. If the surface is not elegant, it is rough surface, if it is having black spots, it always have the fear that it will not consume the tablet. So these are the expected outcomes from the tablet. To obtain this expected outcomes, we have to just add some raw materials, packaging materials to achieve this outcome. So those things, like here I have introduced a slide where it is a difference of uncoated and coated tablets is mentioned. Here very interesting is like this. Whenever we, from this particular slide, I want that our faculty members should understand whenever we teach something, so we should teach in a comparative manner so that it will be easy for a student to understand the things. This particular A type of technology and B type of technology, what is the difference between the two technologies, which is better, and when to use those technology. In an industry, whenever we are taking some project, it is in an industry, we always think first of the cost and the time. These are the two important things in today's scenario, which is very important, like the cost of production. We should produce a tablet which is less in cost. It should not be heavy burden on the cost, because otherwise your net profit will be reduced. So costing is one of the aspects that we have to think of. And the time of the production, like if one batch can be prepared in 1 day and you are preparing that particular batch in 5 days by using your sophisticated technology which you have designed, then that sophisticated technology, whether it is a patented technology, whether you have a patent on that, but if in nutshell, the 1 day tablet and the 5 day tablet is working in having the same bioavailability, having the same stability, then it is useless to make a technology through which the batch can be prepared or will be prepared in the 5 days time. So we have to work on time and cost. So very important thing, like I just make you to understand this thing through this particular slide, which technology we should select, whether like for the tablet, if we are like any project comes to us, so first of all, first question we will think whether we need to prepare the uncoated tablets or coated tablets, right? Then the difference is, if we see from the elegance perspective, coated tablets are always good because they have a very shiny surface. Then they have you can make the color of the coat pink, purple, blue, green, whatever you require, right? But the elegance of uncoated tablet is less. So coating is generally done to hide the spotted appearance of raw materials like black spots or this. But what is in the nutshell, if it does not have the spotted appearance, the uncoated tablet is also very good and the coated tablet is also very good. Then why to go with the coating? Because you should not go with the coating process. Why we should not adopt the coating process? because it increases the cost, because whatever coating materials you are going to use it will increase the cost and the time of production. Like, correct? So unnecessarily, if there is this coating is not functional, is non-functional coating, when there is no use of that particular coating, then why we just only for the appearance that it should look green, blue, red? It should not. We should not coat those particular tablet. Yes, we need to coat the tablets when there is a strength differentiation. In that strength differentiation also, there is no need of doing coating. You can add the color into the matrix itself. Like you can add the blue color, you can add the pink color, you can add the green color when there is a strength differentiation. Like, what do you mean by strength differentiation? If it same tablet coming in 3 different strengths, 10 mg, 20 mg, 30 mg tablets. Like, are you getting me? Like there is certain like cetirizine coming 5 mg and 10 mg. Then you can certainly lower strength can be pink color and higher strength can be green color, so that patient will easily can identify which strength it is. So if generally if it is not functional, there is no requirement of the coating, then you should avoid it. So the students from that particular perspective should know that with this particular angle, because when we prepare our students, we should ask in such a way in our question examination paper by certain case examples, like what we are going to do it today itself. It increases the cost and time of production. Certainly 2 batches of uncoated tablets is equivalent to the time of 1 batch of coated tablets. And moreover, the RM cost increases to 150%. What you can produce in 100%, like 100 rupees, or like you can say 100,000 rupees per batch, it will come to the 150,000 rupees with the coated technology. So certainly you have to think from that particular angle, the cost involved into the coating process. In fact, the time also increases and the cost also increases, right? Then the very next example is the hardness. Hardness if in uncoated tablet is usually less, and coated tablet it is more. And therefore, in uncoated tablets, we generally ensure the friability. As per pharmacopoeia, you need not to do the friability test for the coated tablets. Correct? So uncoated one has the disadvantage that because coating gives the strength to the tablet. That is why one of the reasons that friability is not required. But once it is uncoated tablet, we generally ensure the friability before packaging it, the tablets. Second, uncoated tablets are having the less stability, because coating can provide the stability from the environment, like from oxygen, from water, so that it will not permeate inside the tablet, right? So uncoated tablets are generally prone for degradation, but coated tablets are very much stable formulations, because you can have the oxygen or moisture barrier coatings you can do on the tablets. Like bioavailability perspective, I can say uncoated tablets are high for iron, because it can immediately disintegrate and immediately can highly bioavailable. Matlab, your Tmax will be less, because it will disintegrate and immediately release the dosage form API. But in the coated tablets, you can prepare controlled release and delayed release tablets. I hope my faculty members able to know what is controlled release, where there it is 0 order release, first order release from the tablets. You can able to control the release of API from the tablet. Certainly control release was also be certain time known as modified and extended type of drug release. And DR tablets are delayed release tablets. Delayed release means it's supposed to be release the API into the large intestine or small intestine. Correct? It should bypass the stomach. So it should bypass in stomach. It is the acidic pH. So our coating should be in such a manner. The coating should not dissolve into the acidic pH. And this small intestine and the large intestine contains the basic pH, and we coat with the polymer in such a manner that those polymers should dissolve into the basic pH and release the active ingredient. So this is the beauty of the delayed release tablet. It is also known as enteric coated tablets. It is also known as enteric coated tablets. So bioavailability is generally high if you have the immediate release like the bioavailability concern. Then uncoated tablets are generally preferred. But if it is you need to design the control release tablet or delayed release tablet, then you can opt for the functional delayed release coating material, enteric coated material. Like in industry we use CAP. CAP is cellulose acetate phthalate. Then we use HPMC phthalates. Then we are using, certain people are also using shellac. Shellac is it is a plant material obtained from plant, and it can be used as a enteric coating material. Previously, very primitively, people in 1980s, 1990s, they also use shellac as the enteric coated. It's a natural enteric coated material. So, in the nutshell, I can say if the uncoated tablet should be preferred if API is stable, because if your API is very much stable, it is not prone to degradation by oxygen, by water, moisture contents, then you should prefer the uncoated tablet. And tablets meant for immediate release, then we should prefer the uncoated tablets. So there is no need of doing the coating. Just avoid unnecessary things. We can avoid. So those type of thinking ability we need to develop into the students, where we need to quote and where we need not to quote. That is only possible if we do the comparative analysis between the different type of technologies. Like here I have projected with a very simple example of uncoated and coated tablets. Now we come to the very important thing, critical formulation variables. Whenever we design some formulation, like here we have did coated and uncoated tablet, it is a variable, that coating and uncoated tablet, it is a formulation variable. So before making the formulation, we also, before like preparing the tablet, the intermediate step is the preparation of the granules, right? So how to prepare the granules, what is the composition of granules, like how much microcrystalline cellulose, lactose, how much quantity is required, then porosity of those granules. Like the higher the porosity, higher the bioavailability, you can understand, because the more water can go inside the tablet. If it has highly porous granules, then water will go into that particular granule very easily and can disintegrate those granules also and dissolve the API inside the granule, and it will be release the API into the media very fast. In vivo, the media is our acidic media, that is our gastric fluid or intestinal fluids. It should be released very quickly so that it will be absorbed quickly and give us the response, right? Then size of the tablet is also very important. If it is a size, you are making a very high big tablet size tablet, then it will take more time for disintegration than these smaller tablets, right? The shape of the tablet is very important, because why the shape is very much important? Because if you have the circular tablets, but these are flat tablets, you cannot coat the tablets, because flat tablets cannot be coated. Only the biconvex tablets can be coated, right? It should have some convex on the shape of the tablet. It should be convex, then only we can coat the tablets. Otherwise, the shape is very much important for coating process, because this is one of the critical formulation variable. Whenever you design your formulation and you want the coating, so you cannot select a flat circular punch. You have to select the punch which is biconcave. Punch is biconcave and your tablet will be biconvex. So try to understand this thing, like correct. So in the shape, the surfaces should be biconvex. And density is very much important for the granules itself, because density will select how much is the flowability of the granules or powder, right? So whenever we design some granules, we should optimize the density, bulk density and the tab density. These are the two things which to be controlled so that you get the good flow property of the powder. So there are various things like in industry, like if you ask what is this particular two things is there, then you can understand the angle of repose. Like one of the tests which we generally do for measurement of the flow property, then Hausner ratio and compressibility. These are the three parameters which we generally test to understand the flowability of the powder, right? Now, in the second one, I am talking about type and amount of the polymer coating. What is a type of polymer? Why type of polymer? Because if your API, if your active ingredient is very much prone to degradation from moisture, right? So you have to use a coating material which is not soluble in water and soluble in organic solvents. Because you have to coat through the organic solvent, because if you are going to coat with the water, right? So whenever you are doing the coating in the coating process, your API going to be degraded, right? So we have to prevent the degradation of the active material, and generally in those cases we generally avoid water in the coating material, coating blend, you can say, which you are going to coat on the tablets, right? Or otherwise, if you are using the water itself, the material should not be very hygroscopic. Otherwise, during the stability, during the storage itself, it will absorb the moisture from the atmosphere and it will degrade. So certainly, like there are certain coating materials like HPMC, hydroxypropyl methylcellulose, those to be avoided, because cellulose immediately absorbs water from the atmosphere, and when it absorbs the moisture, it can degrade your API, active ingredient inside the tablet. So you have to use certain material which is acrylate based, like acrylate based, polyacrylate based coatings, so that it will not absorb the moisture from the atmosphere. And now the second is the amount of polymer coating. It is amount. Why we require a particular amount of the polymer coating should be there? Because if the coating is very thin, then also the moisture and the air can permeate inside the tablet. The proper amount of polymer coating is very much required during the coating process. So we optimize that amount of the coating, polymer coating. It should be between, like if we are saying it should be between 3 to 5, means we are optimized that particular, because more than that 5% and less than 3%, the limit is 3 to 5%. What can happen? Less than 3%, the oxygen and water can be absorbed inside the tablet core, right? And if it is more than 5%, like more amount of coating is there, then what will happen? It will not allow the tablet to disintegrate, and it will take more time for the disintegration of the tablet. And once it will take more time for the disintegration, the release of the API, the active, will be slower, right, from the tablet. So you will not be able to match the drug release between the innovator, that is reference product, and your product. So whenever we are making some tablets into the industry, we always compare with the reference product if it is there into the market, or otherwise you will be the innovator one, right? Then the nature and size and amount of tableting excipients is very much important, because if you are using the lot of amount of tableting excipients, then it should be rational. It should not be something like you are using, certain people think if it is contain 9 excipients and certain tablet contain 5 excipients, so which one is better? So it doesn't mean that 9 excipients is there, it is much more better. Always we should make a tablet which is having the least number of excipients, because these excipients can interact with your active, which we generally known as drug excipient interaction. So drug excipient interaction can be prevented if you use the less number of excipients. Otherwise, with our unrational use of excipients can be dangerous for your formulation, correct? And the size of the tablet is very much important. Like as I told you, like bigger the size of the tablet, bigger time to be taken to disintegrate those particular tablet. So this excipient's nature, size is very much important, which I'm going to tell you in my next slide also during the. Is it okay? Some mic is I'm.
Yes, sir. Now is okay, sir.
Okay. Yeah. So this is the critical formulation variables, how to design the formulation. Now we come to the very important stage in design of in the QBD. That is how we design our formulation by the critical material attributes. What are the critical material attributes? Like for any active substance, what are the important points, parameters to be considered for the material? Your active is also a material, your raw materials are also material, then packaging materials is also material, right? So here, the example of API, if you have any API, so the very important material contribute is the particle size. Particle size is not very much important for BCS class 1 or 3 molecules where it is highly soluble, right? But it is very much critical for BCS 2 and 4 molecules where it is a low solubility is there, because lesser the particle size, more is the solubility. So D90 is very much important. D90 means 90% of the particle should will be below that particular particle size. Like if I say D90 of the powder or your API is 50 micron, it means that D90 means 90% of the particles in that particular powder are below 50 micron. Only 10% can be above it. So at least we assure that 90% is below this particle size. So D90 of the particle is very much important if we are making a tablet formulation with the BCS class 2 or 4 molecule, right? The water content in the API is very much important because water can degrade your, like we have to select the starch of those companies only where the MLT is properly controlled. The microbial growth is not there, right? Similarly, the example of the lactose. Lactose is generally used in parenteral formulations also and into tablet formulations also. And the very much important is lactose size, right? Because if you see in the oral disintegrating tablets, we are using lactose, like perlitol, something people are using. It is a particular size of the, like here we are not using the very fine lactose. Here we are using a directly compressible lactose, which we can directly, because oral disintegrating tablets, we generally do not use water. We are making it directly compressible so that it will be disintegrate very fast. It should not have the high binding property. We are not using any binders into the oral disintegrating tablets. And this is one of the reasons we should compressed. We use a lactose of having a particle size of 150 micron to 200 micron so that it can be directly compress. Lactose is also one of the microbial prone excipient that is one of the reasons we monitor the MLT inside the lactose also. And water content is definitely one important thing, because water, if there is more water, it can degrade the API. And then scientists has discovered a new type of lactose which is known as anhydrous lactose. So now many companies are providing anhydrous lactose. There are many suppliers of anhydrous lactose. So water content is, in anhydrous lactose, it is less than 1%. Otherwise, in the normal lactose, it is generally up to 10%. Microcrystalline cellulose is also one of the very highly used excipient into the tablets. And it comes in with different particle sizes. Like the higher the particle size, you can compress the microcrystalline cellulose, and then you need not to have and then the free flow of the blend the granules, these are all can be achieved through the microcrystalline cellulose. And water content, as it is a cellulose derivative, water content and ability is also one of the important tests of this particular material attribute of this particular excipient. Gelatin, everybody knows, like gel strength and pH, because there are two types of gelatin comes, gelatin A, gelatin B. They are different in pH, and pH is very important because if your active is stable in acidic pH and not into the basic pH, then what type of gelatin you have to use into the formulation, it all depends upon the pH of the gelatin, right? PVP means polyvinyl pyridone. Polyvinyl pyridone, because today people are not using much of this starch because it is highly prone to microbial growth. So they are using the binder which is known as PVP, K30. Generally, they are using K90 because it is giving them good viscosity and good granulation. So, but only the problem is like it contains the peroxide. So this peroxide can degrade, it can do the oxidation of your API. So that is why we should use the PVP having the lower amount of peroxide. Or otherwise we should avoid, if the tablet is oxidative prone, we should avoid use of PVP. There you can use the starch. So PVP should be avoided in those tablets where the API is highly sensitive for oxidation. So I generally say don't use PVP, right? In that case, you can use starch. That is a more safer approach to go with the starch. Now, the very important case study which I am going to share with you now is the HPMC. So HPMC comes in different grades depending upon the viscosity, K4M, K100M, right? K200M, right? So K4M is 4,000 viscosity, K100M 100,000 viscosity, that is 1 lakh viscosity. 200M is 2,000, 200,000 2 lakh viscosity. Viscosity of HPMC generally measured at 2% concentration, 2% weight by weight concentration in water at 20 degrees Celsius. So whatever viscosity I am saying, K100M, it means 2% of solution in water is giving the viscosity of 100,000 CPS. Viscosity CPS. Now, here I want to highlight a very important critical material attribute of HPMC, because viscosity is another thing. HPMC comes with the percentage of hydroxy, methoxy, and hydroxy propoxy groups. The HPMC contains, that is hydroxy propyl methyl cellulose. It comes with the different percentages of hydroxy, methoxy, and hydroxy propoxy groups. So this is one of the case studies of my own PhD project. Actually, my PhD project was majorly focused on transdermal, and but one of the projects was of tablet, right? And that very interesting example, I am not going to quote the API, but the very important example of that particular tablet, it was a sustained release tablet. And we designed, that project was of DRL, right? Dr. Reddy's Laboratories. And they have the difficulty in development of that particular tablet, of that particular API. And they then, whenever industry face some problems, they generally give to the academics to solve those problems, right? So in industry, like in academics, we received that particular project from DRL, and I myself work on that particular project. And then we developed it, and with the K100M. K100M is 1000 viscosity methyl HPMC. And we did the technology transfer, right? When it is all like in the pilot studies, like pilot means under the lab studies, and we scale up to pilot level, up to 20,000 tablets into the our own pilot plant, and then we go with the higher scale up to 2 lakh batch size into the industry. And when we went to the DRL, we took this all pilot studies were performed in DRL itself, and it was a very good profile we are getting it. But later on, we scale up to 2 lakh tablets. And then we found there is a drug release, we are not getting it properly. It is a faster drug release we are getting, not the sustained release. The profile which we want for the drug release, it was not there, and higher drug release we are getting. More drug is releasing in the that particular time, that is 2 hours, 4 hours, 8 hours, right? More drug was releasing. Then we investigated, and it was almost took another 6 months to resolve that particular problem, because HPMC, then we understood that from the vendors, that HPMC comes in different grades. Like here I have written 1828. 1828 means the first two digit 18. It is the percentage of hydroxymethoxy groups, and 28 means 28% of hydroxypropoxy groups. So this particular HPMC 1828, it is having the 18% of hydroxymethoxy group and 28% of hydroxypropoxy group. And in the HPMC next is, it is reverse of that. Is here hydroxymethoxy is more, 29%, and 10% is the hydroxypropoxy group. So in the initial development, we were using HPMC 1828, but later on, when DRL invite, they only said K100M, but K100M also of that was a material from Rocket Life Sciences. Rocket is a very big company of Germany. So they supplied the material of 2910. So we in the research we were using 1828, and this is that it was a miss point from us, because during development also we never studied there is a handbook of pharmaceutical excipients, which is their book. If faculties just go through that handbook of pharmaceutical excipients, there in the first page itself of HPMC, they have clearly mentioned that HPMC comes in a different percentages of hydroxymethoxy and hydroxypropoxy groups. But that was missed by me during the PhD, and we make it. And later on, project was successful, but it took time. It was like the time for the industry is very much important, and so I think the literature survey is also very, very important point to be taken care of during the development. Now I come to the critical process parameters during the development. Whenever we design some process, it is very much important. The first process in tableting is denser and calcium is lighter. So what is happening after we mix it, and then after mixing we get a very uniform blend. But during scale up, what we do, don't think this presentation is from that particular angle. From the beginning, I am saying how the different parameters, one parameter can be important for the design of the tablet, because you have to design a tablet, right? So students, it is a responsibility of the teacher itself. They emphasize each and every process parameter, how it can impact. And those parameters you can understand when you make some tablets, and if you're doing some experiment. So generally what we do into the laboratory, what we gave them process, dry at 60 degrees Celsius for 30 minutes. So certainly you break the group into 4 groups and tell them you make a tablet at 60 degree, dry tablet at 60 degree, dry the granules at 60 degrees Celsius for 30 minutes. Tell another group to 70 degrees Celsius for 30 minutes. Tell another group to dry the granules at 80 minutes for 30 minutes. And tell another group to dry at some other temperature. So what will happen? You will be able to understand the impact of temperature on the hardness of the tablet, on the flowability of the granules. So this will help them to understand practically the practical approach, their practical, you can say analytical power will be enhanced. So certainly we have to develop those cognitive skills, that they start thinking on analytical perspective, that if we increase the temperature, what can happen? If we decrease the temperature, what can happen? Correct? Then inlet air CFM and inlet air temperature is very much important, because the CFM is very high of inlet air. What will happen? The granules will dry, and then if the CFM is very high, the CFM means this particular amount cubic feet per meter. This much cubic feet per meter air is blowing inside the chamber, drying chamber. So if the higher CFM, more motion, more movements, and it will what will happen? Our granules will collide with each other and convert into the fines. So it will break down, right? In sizing and milling, the RPM of the blade is very much important, because the RPM is very high. It converts the granules into the fines. Then rate of addition of granules into the RMG, because if you are adding very fast, then what will happen? It will not size properly, and if you are adding very slowly, all the powder will be completely sized and milled, and it converted to the fines. So rate of addition of granules is very much also very important during in the sizing or mill. Whenever you are using some mill, rate of addition of granules into that particular mill is also very much important. In the compression, the very important is the fill volume of hopper. RPM of turret, if it is very uniform, if it is having the uniformity, there is no uniformity issue, you can keep the RPM of turret very high. Otherwise, you have to keep slowly, so that the raw materials and active should not segregate on the turret itself, right, in the cavity. So you have to slowly and you have to keep the speed slow. And if you are if your powder is not having the good flow property, so it will not fill into the cavity, and if the RPM is very fast, it will not fill properly into the cavity, and you will get tablets of lower weight. So that is why it is one of the reasons that we have to control the RPM of the turret. Compression force is one thing, higher the compression force, certain times the tablet becomes very much friable and it disintegrates very properly. So compression force, it should not be too less. The tablet is not having the good hardness, but it should not be too high that tablets, there is an internal cracks inside the tablet and it will disintegrate or break after into the friability test, fail into the friability test. So these are the certainly coating parameters like inlet air temperature, inlet and outlet CFM, pan RPM, and spray rate. Because if the pan RPM is very fast, the tablets you will not be able to coat on the bed of the tablets. Spray rate is very fast, the tablets become moist, and then they will stick together. The sticking problem if you have a very high spray rate. So you have to control this process parameters during the processing of the tablet formulation. Very interestingly. Then what are the final tests which we are going to test? How we are going to control the quality of the tablets? So these are the quality control tests which generally in QC we perform. Like one is the appearance, how the tablet is looking, how it is elegant or not, like the shape, weight. The hardness of the tablet, then friability of the tablet, disintegration time of the tablet, dissolution, assay, uniformity of content, degradation products, and MLT. Certainly MLT, the microbiological aspect of the tablet. So here are the two case studies. Here I want from this as a faculty, this is one of the tests and one of the case study where we can think of to understand whosoever. It is nothing like the first or second, just to understand how we can think of if this is one of the case study, like the basic aim of giving this there are two case studies to use our mind, and you will able to understand this, because if you are going to read the case studies, then it will, these are the type of questions you can give to the students. So therefore, by this type of presentation, only my motive is to how to design the question papers, how to think, how to increase the analytical capability of the. So the answers you have to, because this is only one side talk. So what we can do, you can give the answers in the chat box, and whatever top answers, correct answers, we are going to read it, and then we will find out the correct answers. Correct. So the case study is the API active belongs to BCS class 2 molecule, and it is prone to oxidation and moisture degradation, right? It can prone to oxidation and moisture. Second point is this API quantity per tablet is 5 mg and tablet weight is 220 mg, and it is a sustained release tablet. So these are the three inputs given. So first of all, what will be strategy to get uniformity of content? What will be the strategy to get the uniformity of content? I think I need to, I think you cannot share, or you can write in the chat box. Is it possible? Hello? Or if any faculty wants to speak, like, hello, are you there? Dr. Mehan, are you there? Or hello? Hello?
Yes, yes, Dr. Saab, continue, please.
Yeah. Gupta sir, anybody can write in the chat box? Okay. I think it is better, like I just tell you.
Yeah. Not received any reply. So
Okay.
I hope that this is your fourth lecture today. So most of the from morning 10 a.m. I have started the lecture. First three lectures, they all are basically on the AI.
Okay. So now, okay. So what we can do?
Next fifth one also waiting. So I request you, how much time you have required to complete?
5 minutes, sir.
Okay, okay. Thank you.
Yeah.
Continue, please.
So, okay, I just give the answers. What will be the strategy to get the uniformity of content? We should, because the tablet is only containing 5 mg of the tablet and tablet weight is 220 mg. That is a huge tablet weight because it is a SR tablet, but a lot of polymer is required to sustain the release of the API drug release. So, we should adopt a geometrical mixing strategy to maintain the uniformity of content. What CMAs can be critical, because critical material attributes, so as it is belong to the class 2 molecule, that is a less soluble drug, so the very much important CMA is as the second thing is moisture sensitive. So the water content in the API should be very low, and also the excipients which you are going to use, it should also contain less moisture. You should not use, because it is prone to oxidation and moisture. It can degrade inside with the presence of water. To use select and excipients which contain less amount of water, less amount of moisture. Second example is the API itself belongs to BCS class 2 molecule. So BCS class 2 molecule is less soluble. So there the particle size of API is very much critical. It should not be, we should need to control the D90. It should not be high particle size molecule. API should be there. We need to control the API particle size. Third question, these are the important critical material attributes. Third part is if uncoated tablet to be prepared for bioavailability, because if you are preparing the uncoated tablet for increasing the bioavailability, then what precautions to get a stable product? अगर हमें uncoated ही बनानी है तो we cannot prevent through the oxidation and moisture through coating. It is an uncoated tablet. Then where we need to work upon? We need to work upon the excipients which contain the less moisture. Second, we can have the good packaging material from which the oxygen and moisture will not go inside the tablet formulation. So good packaging material is required. So we certainly need to work upon the packaging material. If design tablet has less bioavailability, then how we can increase bioavailability? I have told you, because if it is having less bioavailability, then you can reduce the particle size of the API to get the more bioavailability. नेक्स्ट क्वेश्चन, व्हाट सीक्यू इज मोर क्रिटिकल, बीइंग एन अनकोटेड टैबलेट? बिकॉज कौन सा सबसे इंपॉर्टेंट टेस्ट है जो कि अनकोटेड टैबलेट्स के लिए रहता है, वो होता है फ्राइबिलिटी टेस्ट। बिकॉज कोटेड टैबलेट्स के अंदर फ्राइबिलिटी टेस्ट नहीं होता, अनकोटेड टैबलेट्स के अंदर फ्राइबिलिटी टेस्ट होता है, राइट? तो दिस इज वन ऑफ द इंपॉर्टेंट क्रिटिकल क्वालिटी एट्रीब्यूट जो मैंने क्रिटिकल क्वालिटी एट्रीब्यूट्स पहले बताए थे, उसमें मैंने आपको बताया था फ्राइबिलिटी टेस्ट है वो अनकोटेड के लिए होता है, कोटेड टैबलेट में हम फ्राइबिलिटी नहीं करते हैं, करेक्ट? सेकंड केस एग्जांपल इज दिस इज एपीआई इज थर्मोसेंसिटिव, वो डिग्रेड हो जाता है हीट पे। पेरेंटल फॉर्मुलेशन टू बी मेड, हमें वी नीड टू प्रिपेयर अ पेरेंटल फॉर्मुलेशन। As we all know, parental formulations are sterile formulations, and we generally do terminal sterilization in autoclave, but it is thermosensitive. So which technology can be used for manufacturing? The answer is lyophilization. Lyophilization is a technology where we can lyophilize at a very low temperature, -°C, and we can dry the powder and we can get a sterile powder. What preservatives can be used to prepare this? No need of any preservative because it is a sterile formulation. So this is the confusion question which I have kept here. You can confuse your students so that they will start thinking which preservative. They write the name of the preservative, but certainly because parental formulations are sterile formulations and no preservatives are required to the sterile formulations. So in this summary, I can say comparative analytical capability to be developed among students. Some case study examples can be incorporated in practical or theoretical exams. I hope you must have understood this lecture and the purpose of this lecture. Thank you very much.
Thank you, sir. So I request to Dr. Kamya to
countries and territories with the top 5 destinations being the USA, Belgium, South Africa, UK and Brazil. Next factor is global presence. The industry boasts a strong network of over 3,000 drug companies and about 10,500 manufacturing units. It is the world's 3rd largest by volume and 14th largest by value. And what is going to be the market size in coming years? The projection is of 120 to 130 billions by the year 2030. Currently, it is 60 billion USD market. So the Indian pharmaceutical industry is poised for continued growth both in India and worldwide in the coming years. Do you agree with this?
Yes, ma'am. Yes, ma'am.
Yeah, next slide, please. So here are the few government initiatives to sustain the growth, this huge growth which is going to happen in coming years, and this is the Vision Pharma 2047. So what it says, make India a global leader for affordable, innovative and quality pharmaceuticals and medical devices for the goal of Vasudhaiva Kutumbakam. Vishwa Guru in innovation and research for delivering healthcare products to future generations in a sustainable manner, introducing natural products also. Ensure accessibility and affordability of patient-centric products for better healthcare by building upon partnerships across industry, science, and government. Create equilibrium between social, economic, and governance aspects through facilitative, balanced, and progressive policy and regulatory framework. Reduce India's carbon footprint in pharma medical technology to align with Honorable PM's vision of Panchmitra. Identifying critical suppliers, de-risking the supply chain and promoting local sourcing. Medical devices to be an integral part of global supply chains for raw materials, components, spare parts, assemblies and sub-assemblies. And last is digitization, technology upgradation in the delivery of services and products under Jan Aushadhi Pariyojana. So these are the few government initiatives which are being taken by the Government of India to meet the pharma market for 2030, and this is the Vision Pharma 2047. Next slide, please. These are the few schemes which are being run by Government of India. The Government of India has launched several schemes to promote industry-academia linkage. These schemes encourage collaboration through joint research projects, technology transfer, and industry participation in curriculum development. By fostering closer ties between industry and academia, the aim is to accelerate innovation, enhance the competitiveness of the Indian pharmaceutical industry, and ultimately improve healthcare outcomes. So this is a list of a few initiatives like research-linked incentive schemes. This rupees 5,000 crore scheme aims to strengthen research infrastructure and promote industry-academia linkages for research and development in the pharmaceutical and medical technology sectors. Next is PREP, which is a scheme for promotion of research and innovation in pharma medical sector. This scheme promotes collaboration between the private sector and government institutions, focusing on priority areas to boost India's position in the global pharmaceutical market. Next is National Policy on Research and Development and Innovation in Pharma Medical Sector. This policy aims to create a world-class research environment, develop a talent pool, and promote industry-academia linkages. Pharmaceutical Technology Upgradation Assistance Scheme, PTUS. This scheme facilitates pharmaceutical MSMEs with proven track records to upgrade their technology. Next is the PLI scheme for pharmaceuticals, which provide incentives to promote domestic manufacturing of pharmaceuticals, including those developed through industry-academia collaboration. So these are the few initiatives which are being taken for industry-academia linkages. Next slide, please. So this is about the various industry-academia linkage centers which are being developed by government in upcoming bulk drug parks to increase more and more interaction between academia and industry. So what is the purpose of having these centers is creating conditions that improve the interactions between industry, academia, research labs, and students, as well as skill upgrading. Creating mechanisms that will increase the level of structured innovation and entrepreneurship so that more students from becoming job creators rather than job seekers, especially in the API sector where the state has a comparative advantage. This is I'm sharing this center for which is upcoming bulk drug park in Himachal Pradesh state. This is going to happen in coming years. Next, please. So in this slide, we are going to discuss about what is the current academic scenario. The current academic scenario for private pharma colleges in India is dynamic and evolving with a mix of positive trends and challenges. Here are the few positive trends. First is the growing demand. The pharmaceutical industry in India is expanding, leading to increased demand for skilled professionals. The growth potential we have already seen in our previous slides, right? So as the growth is going to happen in coming years, so more and more skilled professionals will be required by the industry. To fuel this growth of this fuels the growth of private pharma colleges. So they have to play a key to generate and to upgrade the skills of the students to make them industry ready. Next is diverse course offering. Private colleges offer a range of programs including B.Pharm, M.Pharm, PharmD. These are the special different courses which are being offered by the colleges, and specialized courses in areas like pharmaceutical analysis, quality assurance, and regulatory affairs. Third is industry collaboration. Many private colleges are focusing on collaboration with pharmaceutical companies for internship, training, and placements, enhancing practical exposure for students. Focus on research. Some private institutions are investing in research facilities and encouraging students and faculty to engage in research activities. Adoption of technology. As everyone is aware, technology is advancing day by day. There is an increasing use of technology in teaching and learning, including online courses, simulations, and e-learning platforms. Next, please. So what are the weaknesses for current academic scenario? In this current academic scenario, which is no doubt which is evolved through the years as per the industry requirements, but still there are few weaknesses which we need to bridge upon. First is the variable quality. The quality of education varies significantly across private colleges. Some lack adequate infrastructure, qualified faculty, and research facilities. Emphasis on theory. Many colleges still focus heavily on theoretical knowledge with limited practical training. This leaves graduates ill-prepared for the demand of the industry. Limited research focus. Research is not a primary focus in many private colleges, which hinders the development of innovation and advanced skill in students. Next is placement challenges. While some top private colleges have good placement records, many struggle to provide adequate placement opportunities for their students. So overall, private pharma colleges in India have the potential to generate a skilled pool for the pharmaceutical industry. However, there is need to focus on how to unlock that potential. To unlock that potential, our faculty need to play a major role in that. Next, please. Next slide, please. So what role faculty can play to meet the requirements? Although the role is very crucial in generating skilled pool to meet the growing industry demands, here is the few initiatives which first is the curriculum enhancement. This is industry aligned syllabus. Regularly update the curriculum to reflect current industry practices, technology advancements, and regulatory changes. Include subjects like pharmaceutical data analytics, artificial intelligence in drug discovery, and advanced manufacturing techniques. Next is focus on practical skills. Emphasize hands-on training through laboratory work, workshops, and simulations. Incorporate real-world case studies and problem-solving exercises to develop critical thinking and decision-making skills. Second initiative is industry interface. What we can do here under this platform is collaboration and partnerships. Establish strong ties with pharmaceutical companies, research organizations, and regulatory bodies. Facilitate guest lectures, industrial visits, and collaborative research projects to expose students to the industry environment. Internships and apprenticeships. Arrange mandatory internships for students in reputed pharmaceutical companies. This provides valuable practical experience and helps students understand industry expectations. Move to next, please. Next slide. Yes. Skill development. Soft skills training, conduct workshops and training programs to enhance communication, teamwork, leadership, and interpersonal skills. These skills are essential for effective collaboration and career advancement in the industry. Technical skills enhancement provide training on advanced analytical techniques, instrumentation, and software used in the pharmaceutical industry. This ensures that students are well versed with the latest technologies and can readily adapt to the industry environment. Next is the research and innovation. Promote research culture. Encourage students to participate in research projects and present their work at conferences and seminars. This fosters a spirit of inquiry and innovation, which is crucial for the growth of the pharmaceutical industry. Is everybody agree on the role of faculty?
Yes, ma'am.
Next, please, then. This is entrepreneurship development. What, like, from how we can make our students from job creators rather than job seekers. Incubation centers and startup support. Establish incubation centers and provide support to students who wish to start their own pharmaceutical ventures. This can help create new businesses and generate employment opportunities in the sector. Entrepreneurship training. Conduct workshops and training programs on entrepreneurship, business planning, and financial management. This equips students with the skills and knowledge needed to succeed as an entrepreneur. So by focusing on these areas, private pharmacy colleges can play a pivotal role in shaping the future of Indian pharmaceutical industry and ensuring that it has steady supply of skilled professionals. Go to next, please. Next slide, please. Yeah. So this is faculty development. How the faculty can play a major significant role to create industry-ready students. To ensure faculty members stay in the forefront of research trends and industry practices in the pharmaceutical sector, a multifaceted approach to faculty development is essential. Faculty को भी हमें develop करना है. अगर हमें students को develop करना है तो हमें faculty को भी faculty development भी उतनी ही जरूरी है. So here are some key practices where we can invest in our faculty members. First is the continuing education and training. Workshops and seminars. Organize regular workshops and seminars on emerging areas like drug discovery, nanotechnology in pharmaceuticals, personalized medicine, artificial intelligence in healthcare, and regulatory affairs. Online courses and webinars. Utilize online platforms to offer flexible learning opportunities on specialized topics, industry-based practices, and technological advancements. Certification programs support faculty in obtaining relevant certifications in areas like clinical research, quality assurance, and pharmacovigilance. Next is industry engagement and collaboration. Industry internships facilitate faculty internships in pharmaceutical companies to gain first-hand experience of current industry practices and challenges. Students के साथ-साथ faculty का भी internship program होना चाहिए. Guest lectures and industry visits. Invite industry experts to deliver guest lectures and organize visits to pharmaceutical facilities to expose faculty to the practical aspects of the training of the industry. Next. So this is curriculum development enhancement. How faculty plays role in this? Regular curriculum review. Conduct regular review of the curriculum to ensure alignment with current industry needs and emerging trends. Incorporating case studies and real-world examples. Encourage faculty to incorporate relevant case studies and real-world examples from the industry into their teaching. Integrating technology in teaching. Promote the use of technology in teaching and learning, including online simulations, virtual labs, and data analytics tools. Next is faculty exchange programs. these are both at national and international level. Establish exchange programs with other universities and research institutions, both nationally and internationally, to facilitate knowledge sharing and collaboration. Mentoring and professional development. Mentoring programs pair junior faculty with senior faculty or industry mentors to provide guidance and support in their professional development. Leadership development programs offer leadership development programs to prepare faculty for leadership roles in academia and industry. Performance evaluation and feedback. Implement a system for regular performance evaluation and feedback to identify areas for improvement and growth. Next. This is a faculty role and project designing. This role is very crucial. Faculty plays a crucial role in project conceptualization for students that are relevant to industry practices in the pharmaceutical industry. First is project conceptualization, real world problems. As I shared in my earlier slide also, develop project ideas based on real world scenarios and challenges faced by the pharmaceutical industry, such as drug discovery, formulation development, QC, regulatory compliance, and supply chain management, and others too. Industry collaboration, industry mentors, like in projects, they may incorporate professionals from industries as a mentor to guide the students for their projects from industrial point of view. Facilitate mentorship programs where industry professionals guide and mentor student project teams, providing valuable feedback and insights. Project supervision and guidance, critical thinking and problem solving. Encourage students to think critically, analyze data, and solve problems using industry standard methodologies and tools. Assessments and evaluation. Industry relevant criteria evaluate student projects based on industry relevant criteria such as technical accuracy, feasibility, innovation, and potential impact. I hope after incorporating these initiatives by the faculty while conceptualizing projects for the students, we can meet the goal. Other initiatives which a faculty can take to help students develop soft skill essential for success in the pharmaceutical industry. Some key areas, these soft skills are equally important for growth in the industry. So first is the communication skill. Effective presentation trains students to present complex scientific data clearly and concisely, both orally and in writing. This includes data visualization, report writing, and delivering compelling presentations. Scientific writing develops skills in writing research papers, regulatory documents, and other technical reports with precision and clarity. Teamwork and collaboration, interdisciplinary collaboration. Create opportunities for students to work in diverse teams, simulating cross-functional collaborations common in the pharmaceutical industry. Conflict resolution equips students to find mutually acceptable solutions within a team. Team dynamics, foster understanding of team roles, responsibilities, and effective strategies for achieving shared goals. Problem solving and critical thinking, decision making, make them decision makers. Provide opportunities for students to make informed decisions based on available data and consider potential risks and benefits. Adaptability and flexibility. Prepare students for the dynamic nature of pharmaceutical industry where new technologies and regulations constantly emerge. Time management helps students develop effective time management skills to handle multiple projects and meet deadlines in a fast-paced environment. Time management is very important. Resilience, foster a growth mindset that encourages students to learn from setbacks and adapt to changing circumstances. Ethical and professional conduct, integrity and accountability emphasize the importance of honesty, transparency, and ethical decision making in all aspects of pharmaceutical work. Professionalism instill professional etiquette, communication, and behavioral expected in the industry. All these are the soft skills which faculty should focus on to develop among students. These are very helpful for the students to get ready for the industry job. So, to summarize today's presentation, what we talked about teaching transformation is a process that goes beyond simply imparting knowledge. It aims to empower learners with skills, values, and mindsets that enable them to thrive in a rapidly changing world. From passive to active learning, traditional didactic methods are giving way to active learning strategies where students are engaged in problem solving, critical thinking, and collaborative activities. From teacher-centered to learner-centered, focus is shifting from the teacher as a sole source of information to learner as an active constructor of knowledge. From content delivery to skill development, all these are the changes which we are looking for a change. Like faculty has to play a major role here to make these changes in their lecturers. Emphasis is placed on developing 21st century skills like creativity, communication, collaboration, critical thinking alongside content mastery. So this was all about today's session. I hope I met your demands and requirements. My deepest thanks for your consideration and good luck for your future endeavors. Thank you so much.
Thank you, ma'am. Thank you so much. I request to Dr. Kamya for proceed the session further.
Thank you, ma'am, for such a valuable session. Now, I would like to invite our worthy director, Sir Dr. G.D. Gupta, sir, to conclude.
So thank you very much, Dr. Sivaliji. First of all, I congratulate to you for starting one of the new platform. This is the clap, and I hope that it will be the very, very industry and various you can say rules and regulation, as well as the what types of the things is the thinking about the government of the India, as we all are awaiting over here 2047. We are going to, after the completion of the 100-year century, where we are, where our pharma industry, where our education. So everybody preparing most probably toward this region, and you have considered each and every point very specifically, scientifically, and I hope that this talk it will be the very, very helpful to the teachers. Today is the grouper, so but they all are involved and connect with the all these of, you can say, these are the lectures. So madam, I just want to give over information. Here is the more than 40 participants. They are the physically registered on this platform as a offline mode, and 2025 registered on the online mode. So your voice is directly live to up to the more than the 2000 peoples, and this lecture also available on the platform of the ISF College of Pharmacy YouTube channel. So I hope that
Thank you, sir.
everybody will take the benefit, and I also request you can send the detail of your platform and how we can connect and how it will be the helpful to the teaching learning process, as well as how it can be connect to the industry. So thank you very much.
Thank you,
And
sir. Thank you.
Thank you, ma'am. Thank you, sir. So there's an end of 4th day FDP. Hope you all understand and get knowledge and benefits from today's sessions. Now we gather together by tomorrow sharp at 10 a.m. Thank you. Thank you, everyone.
Thank you so much. So I request to faculty members, they can left the group.